Good afternoon. Welcome, everyone. It's my distinct and special pleasure to welcome you to our 10th annual Rare Disease Day event here at Broad Institute. And I would like to first welcome my co-chair for this event, Belinda Termeer, to say a few welcoming remarks. Thank you all for coming.
Thank you. So hello, everyone. My name is Belinda Termeer, and I'm the president and co-founder of the Termeer Foundation. It is my honor to welcome you today to an era of innovation and to congratulate the Broad on the 10th anniversary of hosting this Rare Disease Day event. I'd like to start by thanking the Broad Institute for their partnership on this event, specifically Drs. Anna Greka and Jillian Shaw. The Termeer Foundation is honored to continue collaborating. So innovation is at the center of our work at the Foundation. I look around, and being surrounded by exceptional entrepreneurial thinking leadership is an honor. Thank you to all of you, to today's speakers and everyone in the audience for your commitment to patients, this industry, and the world. The Termeer Foundation was founded to continue the legacy of my husband, Henry Termeer, who for many was an inspirational leader. Our goal is to connect life science innovators, biotech entrepreneurs, CEOs, and visionaries because we believe that helping leaders succeed will ultimately help their innovations reach patients. We believe in the work of everyone in this room and are honored to play a role in amplifying the voices of rare disease researchers, discoverers, and CEOs. We recognize how important it is to foster community and collaboration in this space. Days like today remind us to pause and come together. With that, we are inviting everyone to join us in partnership, skill, and knowledge sharing towards driving impact and change in the lives of rare disease patients and families. Again, thank you for joining us.
Thank you, Belinda. One couldn't ask for a better partner and co-chair for these events. Our two organizations, the Termeer Foundation, Catherine Smith, who helps run that together with, on our side, Jillian Shaw, and then also Katie Liguori, and others, I'm sure, on the Termeer Foundation side, who I haven't mentioned, are very much the people behind this event today. So I want to make sure that we don't forget, sometimes it happens until the end, but to acknowledge them at the beginning and say a great thank you for bringing us all together today for this special event. So my name is Anna Greka. I am a professor at Harvard Medical School. I am a core institute member here at the Broad Institute, and I have really dedicated my scientific career to understanding rare diseases and hopefully building therapies for rare diseases. And it is actually our 10th anniversary today, the 10th event that is being organized here at Broad, which really coincides with a conversation I had about 10 years ago, or 10 and a half years ago now, with the founding director of the Broad, Eric Lander, and then subsequently with the current director of the Broad, Todd Golub. In both cases, I sort of made the case for how can the world's, or one of the world's premier genetics and genomics institute not have a special day for rare diseases. And of course, in the typical entrepreneurial spirit of the Broad, they said, well, why don't you start one? And so here we are, 10 years later, celebrating the 10th anniversary of this day of coming together to talk about rare diseases, to meet with all the stakeholders in this broad community. It's the scientists, the families, the patients, the physicians. Everyone is welcome under this big tent. And it really, in many ways, affects all of us in different parts of our lives. As you know, of course, rare diseases, there are about 10,000 rare diseases. They disproportionately affect children, of course, and it is a big fight to try to understand them and develop therapies for as many of these diseases as quickly as possible. And many of you and many of the people we'll hear from today are very much engaged in this fight. And of course, one has to acknowledge at an event like this that there are some headwinds that we're all facing at this time. I think it's important to mention that there have been, there was a lot of hope for the renewal of the priority voucher review program, for example, which many of our colleagues in biotech and pharma would have relied on in order to make investments and make progress in this space. And, this is something that did not happen in December. And so it's something that we now hope will become part of the conversation again. And of course, we all, wish to advocate for that program to be reinstituted so that it can help our patients, especially our pediatric patients with rare diseases. And I think this generally compounds an overall landscape that one, again, has to acknowledge in biotech and pharma, where there has been some reluctance in making investments in this space. And again, I think that we all, understand this, and we all feel it in different aspects of our lives. But as everyone who knows me knows, I am an optimist and a glass half full type of person. And so I couldn't be more excited about what this next decade will bring in terms of our abilities to address rare diseases, especially an area that our group here at Broad is very much interested in, rare genetic diseases. We have launched an initiative that is really focused on making progress in this space. We call it the Ladders to Cures Accelerator. Some of you may have been to our symposia. We have another one coming up in May, which you're all absolutely invited to attend. But the whole concept of the Ladders to Cures Accelerator is to actually, in simple terms, try to harness what we understand about the biology of rare diseases in a massively parallel fashion so that we can find nodes, targetable nodes that underlie many diseases, so that we can potentially develop treatments for as many of these diseases as possible, de-orphan as many diseases as possible. And so you will hear today from some of the speakers who are involved in the Ladders to Cures Accelerator, because they have received support from our accelerator to push some of the what we call nodal biology projects forward. And you'll hear about progress that's happening in that space. You will hear more of that at our annual symposium, which is happening in May. But there's also some deep verticals that we're fighting for as part of this project, this accelerator, which basically means that we're going deep into understanding nodal biology here at Broad by really attacking simultaneously thousands of rare genetic diseases modeled in human cells in parallel, and interrogating them with modern tools like CRISPR technologies and AI-driven imaging approaches that allow us to actually understand about the underlying biology of thousands of misspellings in the DNA in a massively parallel, simultaneous fashion. And no one could be a better speaker to represent some of those efforts that are happening here at Broad than our keynote speaker for today, Dr. Anne Carpenter, who is, of course, my colleague, my friend. She's an institute scientist here at the Broad, and she's really a pioneer in what you hear now more and more, like AI-driven image processing and understanding images. Well, Anne was doing it way before this was actually cool or a buzzword that everyone used all over the world. And I think you will hear something about all these foundational discoveries that she made possible, which now make it possible for us to potentially understand thousands of rare genetic diseases in a massively parallel fashion. So without further ado, I will let Anne take it from here. Thank you so much for coming.
Thank you so much. I'm so proud to be part of this celebration today of how far the community of rare disease research has come, and also pushing towards what's in the future. So really excited to be part of this event today. Thanks for organizing. Thank you, Belinda, as well. I want to begin with just this visual that you may have seen before of all the individual genome regions associated with disease that have been discovered. Back when I started my career, there weren't so many, and now it's just exploded. This doesn't even come up to present day. And what this chart tells me is that while, of course, it's wonderful if we could assign a team of 1,000 researchers to each disorder out there, it's just not going to be scalable, even to keep up with the new diseases that are discovered every day through genome sequencing, much less to get to this backlog. So we need something more systematic, and of course, we always think, well, technology should help us here, right? And there are some amazing technologies that have come into play. Computing is one that I'm very excited about as a machine learning and computational biology expert. You might be familiar with this view, Moore's law, which is showing on a log scale this sort of exponential decrease in the cost of compute over time. And it's just a phenomenal peak of human ingenuity that has allowed us to stay on this track and allow computing to be faster and faster, better and better to accomplish so many things, including in biology. And as you probably well know, sequencing has gotten cheaper and cheaper. It used to cost $100 million to sequence a genome, and the reduced costs of this have even broken Moore's law, even better than computing over time. And so you think, okay, well, this is great. Technology is great. Let's do the same for drug discovery. And it at first looks like an awesome picture, but then you notice, wait a second, the scale is wrong and upside down here, because instead of looking at at the cost of a drug going down and down over time, instead what we're looking at here is exactly the opposite. That in the 1940s, you could get 30 new drugs if you had $1 billion, even in today's dollars. But now it's somewhere between $1 to $6 billion per drug. And this is absolutely not scalable to rare diseases or even the more common ones that we have. So we call this a Room's law because it's Moore's law backwards. And it's something that really needs to be fixed. And why is it so expensive? Why has it gotten more expensive over time, even though we have all these amazing molecular techniques, cell biological techniques, computational techniques, and yet it's getting more expensive over time? The answer is, first of all, there are just a lot of steps in drug discovery. This simplifies it quite dramatically, but there's hundreds of steps along the path from having an idea of what might cause a disorder, what are the proteins involved, what are the pathways involved, to then finding a small molecule or another type of fix for the disorder, and then all the testing in cell systems, animal systems, and ultimately in humans for clinical trials. But I would say really that's the time element. The cost element really comes down to this, that drug discovery fails most of the time. And so even with everybody's best efforts, all of our ingenuity, all of our hopes and dreams going into drugs that go into human clinical trials, 90% of them fail. And things just don't work the way we expect, because humans are a very complex system, and chemistry doesn't always work out for small molecules. Gene therapy doesn't always work out. And so this is the real struggle of why it's so expensive to cure illnesses. Okay, but maybe robots will come to our rescue, or at least AI and machine learning. You may have seen a lot of headlines about the new advancements, and there are many, and they are very exciting. I don't know that they're going to reverse Room's law in the upward trend, but even if we could just stabilize, we would be quite delighted. And it turns out that a lot of these pharma companies that have been built from the ground up with some kind of a basis of using large-scale data and using computational techniques, they are starting to get drugs into clinical trials. It's too early to say whether these will be dramatically different than and faster in the preclinical stage, or whether the success rates will be different. There's just too few that have actually gone to human clinical trials, but there's at least some inklings on the horizon. So today, what we'll talk about is not the entire field of AI and drug discovery, but rather some very specific areas where I'm seeing progress happening in drug discovery, where machine learning together with beautiful kinds of data and clever new molecular and cell biology techniques are allowing us to make some progress. So that's what we'll talk about today, is how we use images. So you might all be thinking a lot more, if you're in the rare disease space, you might be thinking a lot about genomics and molecular proteins, genes, mRNAs. But here I'm talking about images, and I'm literally talking about microscopy images of cells. It seems a little bit of an old school kind of technique. But because computational methods allow us to extract information from images and turn those into numerical data, we can then do a lot of the same kinds of things we do with molecular data. So there's two big things I'll talk about today. The first is measuring known phenotypes in images, by which I mean measuring things we know we want to look for in images. And then the second half, we'll talk about profiling to characterize samples. So I'll explain what that means as we go along. So first off, measuring what we know we want to measure. When we are looking at images of these particular cells, here we're just highlighting the DNA of the cells. So these are nuclei that we're looking at. And you can think about how to cure cancer, in this case, this type of AMKL. One way is to kill the cancer cells. Great, if you can do it. Another approach, though, when that doesn't seem to be working out so well, is to just cause the cells to sort of stabilize, just stop proliferating. That alone would be good enough to solve many types of cancer. And so the researchers we worked with here, John Crispino from Northwestern University led this work with a whole team here at the Broad. they thought, well, let's see if we can find some compounds that push these cells into this polyploid state where the DNA has divided, but they've just kind of stabilized. They don't proliferate as quickly anymore. And examples of what this looks like is shown here on the right. And we used, in this case, image analysis software to measure what they knew they wanted to look for. We want nuclei that are big and have a lot more DNA in them. And so that's what they did in this experiment. Very simple kind of readout from an image analysis perspective. But it allows us to screen tens of thousands of drugs here at the Broad and identify that one that was already being used for other sorts of cancer looked really promising. And they ended up deciding to test it in a clinical trial for myelofibrosis, which was successful. So this is one of the first forays here at the Broad that led to a successful clinical trial. And again, we're looking at the images and we're measuring a thing that we already know is a useful outcome for patients. And it worked well here. Leukemia is a nice thing to study because you can take blood from the patients and do a lot of experiments with it. It's very kind of native context, so that worked out very well. But now let's turn to this second kind of way to use images, which is profiling. So what if we don't know what we're looking for? What if we're working in a disease area where we don't know the genes involved, we don't understand them, maybe we know the gene, but we don't really understand the mechanism of how that causes a disorder in the human whole organism? In these cases, we've really found a lot of use in this strategy called profiling. And what I mean here is instead of measuring just the one thing that we care about in the images, like how much DNA is present, instead we measure everything we can about the cells, and then we use that numerical data kind of like a barcode or like a fingerprint of what is happening to the cell when this disease is present. And this strategy is called image-based profiling. We often use something called the cell painting assay. I won't describe today, but that's the assay that labels the cells. And we can think of an analogy here to human patients. So to many of us, we can look at a lot of little kids and have no clue whether they might have some kind of underlying pathology. But for pediatricians who specialize in many of these different kinds of disorders, they have a very visual, very strong ability to detect a disorder just based on a picture of a human face. And so it turns out, of course, computers are pretty good at that too. If you give them a training set and explain that these children have Down syndrome, these children have Angelman's, they are able to distinguish these just based on the facial structure features alone. So what we're doing here with image-based profiling with cells is the exact same thing, just sort of zooming in quite a bit. So of course, you know, your dermatologist can tell if your mole is sketchy looking if they look at a picture of it. And your pathologist can take a slice of a potential cancer and take a look at it and see what's going on. All of these are examples where the physical structure of the thing tells us whether there's a disease. So let's take that concept and use cells growing in a dish, which are cheap and easy to acquire, and most importantly, we can perturb them with tens and thousands of different drugs. We can knock out all their genes and see what happens and so on. They're much more manipulated in the dish, and yet we can still take pictures and see what's wrong with these cells. When we knock out this gene, do they look a little funny, and so on. So this is the field of image-based profiling, and what are some of the things we can do with it? The first example I'll give was originally an academic project. I take no credit for this experiment other than they used our software in order to extract the features that they needed for this project. So what was it? At the University of Utah, they took some cells and grew them in a dish and stained them with some stains. And then they knocked out a gene that was known to cause cerebral cavernous malformation, CCM. And when they made that knockout, lo and behold, it had an extremely obvious phenotype. You can tell something's wrong with these cells. They're much more green. The red stuff is much more bright. And what that means is now you can take cells that are in this unhealthy disease-associated state and test drugs systematically. Can I find a drug that makes the unhealthy cells look healthy again? And what's lovely about this kind of an experimental setup is these researchers happen to be experts in CCM, but you don't actually have to be an expert. Just knock out the gene that causes the disorder and look at the cells. Is there something wrong with them? And you can look at them, of course, by eye, which they did originally here, but you could also look at them with a computer to get much more subtle effects. If it had been more subtle, maybe you'd need a computer to detect this. Now in this case it was obvious, and what they did is they chose some drugs out of this big screen after doing this experiment. They examined every pair of images by eye, and they also let the computer pick some of the drugs to follow up on. It turns out the ones that the computer picked were actually better than those chosen by the experts, and eventually led to a drug which is now in phase 2 clinical trials as part of Recursion. So they spun off this company, and this was how this whole company got started. They now have hundreds and hundreds of these kinds of phenotypes because this is a very systematic thing. You don't have to study each disease for decades. You just, if it's a loss of function kind of disease where the gene function can just be perturbed, we can just see, is there a phenotype? If yes, screen a bunch of drugs to fix it. And that's the systematic approach that they're taking within this company. They now have eight clinical trials in progress. Okay, so back to my lab, we realized this is really powerful to be able to analyze images in a systematic way. And so about 5 years ago, I helped to organize this large consortium consisting of the number of pharma partners that you see here, as well as a lot of academic collaborators and other service providers in the biotech space. And what we did is we created a giant data set, which is now available, and you can see the website and what we've produced here. But what's really lovely about it was we've taken cells and we've said, let's just create one baseline data set that has lots and lots of genes that have either been knocked down using CRISPR or overexpressed, and also a lot of different small molecules. And we can use this resource for all kinds of different things. I'll just highlight a couple of them today. The first is to just learn more about the gene or the chemical perturbation. So there's 20,000 genes in the human genome, and weirdly enough to me, if you had asked me 20 years ago, I would have thought by now we would have a decent idea on the function of most of them, but many have 0 function annotated. Some of them have a function that's been described, but it's not the whole story. And so what's lovely about this data set is you can go to the website, type in your gene of interest, and find out about the impact of either overexpressing or knocking down that gene in this given cell line. What do the cells look like? Are there other genes that produce similar kinds of phenotypes? We've used the term nodal biology today. Maybe I should define it before we go too much further. That if it's the case that every single variant and every single gene is completely different and completely unique, and we have to solve them all one-to-one, hand-to-hand combat, it's going to take a very long time. But what if it's the case that multiple variants have actually kind of the same impact on protein function, or multiple genes, when they're perturbed, have kind of the same impact on an overall pathway? It turns out there's certainly many examples where this does happen, and if we can, step one, group all of the variants or the genes into some categories, some nodes, and find some commonalities amongst them, then we have a better chance of dividing and conquering. And so that's one of the ideas here is that if you type in your gene, you find other genes that are similar to it that might give you some clues. Okay, maybe my gene has no functional annotation, but if its impact on cells is exactly the same as some other gene that is annotated, it gives me a bit of a clue on which way to go. Or maybe the morphological features tell me something. In this one, the DNA has a funny texture, or in that one, the mitochondria are located in an unusual location, and so on. The other cool thing we can do when we have a giant database like this is we can start going hunting for drugs. And so let's say I have a gene that I know is associated with a human disorder, and I perturb it, and I do see a phenotype, like I described in the case of Recursion in the beginning. So once we see that it has a phenotype, I can say, in this database of all of these cell images that I've created, have I ever seen a drug that produces that same kind of impact or the opposite kind of impact on cells? And by doing this matching exercise, it's a pretty straightforward computer vision approach, we are able to filter down all the hundreds of thousands of compounds or genetic perturbations and look for matches. So we did this in one experiment where we had 30,000 profiles to match against, and we typically picked 20 or 30 drugs to follow up on that looked like they would impinge on the gene of interest. And sure enough, it doesn't work all the time. It would be kind of a miracle if it did, because this step usually costs a few millions to tens of millions of dollars to run a high throughput screen to physically test compounds to determine whether they have some kind of impact that you're looking for. Instead, we're taking that 1 year and $10 million and doing a computational query in an afternoon. You don't expect it to work all the time, but we were shocked to find that it worked 32 to 42%, depending how you count exactly. And you can read the paper for more details. One particular case, we used YAP1 as the query gene because we knew it was important in a rare type of cancer called sarcoma, and identified some drugs that looked really promising for it. So promising that we spun off a little company to get started at trying to attack these types of cancer and solid tumors. Okay, so now we're getting really excited about scaling things up. If we can gather large sets of images, we have the potential to solve lots of disorders in parallel. And so we see this vision of a future here at the Broad, largely led by Anna Greka, of this variant to function pathway where, okay, sequencing, we've got that down pat and we're very good at it, and it's a lot cheaper than it used to be. So we can find variants that are associated with disorders, but then how do we go from there to drugs? And one way is through this kind of profiling. I am excited about profiling with morphology because it's very inexpensive and very scalable kind of technology, but we can also use transcriptomes and proteomes in order to capture all the information. When this change in the DNA code is there, what do I see as the impact on the cell? And by doing this, we can do two kinds of things. We can immediately gain phenotypic insights, like the DNA looks funny, the mitochondria looks funny, the mRNA levels look odd. But we can also classify these variants, and that's where we get to the nodal biology. We can find the nodes that group a lot of different variants or different genes with each other. And that allows us to either find diseases and try to reverse them with drugs, either computationally, like I described, or through physical experiments, or we can kind of go the old-fashioned way and really dive into each disease once we know a little bit more about its mechanism. So my last project that I'll describe is a very large one. It's called Project Verchamp, and our goal is to, as you can see, a very large group of people are involved in this project. I'll just talk about a tiny bit of it today, which is that the big picture is taking 80,000 coding variants. It's not every coding variant that exists in humans, but it's in a lot of them. And you can see some of the details here. Some are known to be benign, some are known to be pathogenic, and some are uncertain. And we're working to get these all cloned and tested, and what are we gonna do with them once we have them? We're gonna measure all the things about them. So when this variant is present, what happens to the cells? There's other parts of the project that are working on testing, does this variant cause the protein in which the variant exists, is it causing that protein to interact differently with other proteins? The second one is, when that variant is present, does the amount of the protein change? Does it knock it out completely, for example? But we'll talk today about the localization and morphology, since that's what my lab has been working on directly. Does this variant have any impact on the localization or the morphology of the cells? So here, it's a collaboration with Mikko Taipale, who did a beautiful experiment with his graduate student, Jessica Lacoste. And here's one example of how this works. You take a protein that's known to be associated with a human disorder, you express it in cells and tag it so you can see it. And so here's 3 cells, 3 kind of dark oval nuclei are not shown in the middle, and then the green stuff is around it. And then you just say, well, if I introduce this variant that's associated with Noonan syndrome, does that localization change? And I don't have to know anything about Noonan syndrome or this protein or its normal function or even what these dots are. I can see there's a difference. And then all I have to do is test a bunch of drugs to see if I can find one that reverses it. So in the same way that a pediatrician, I wouldn't notice that there's anything unusual about this boy, but a pediatrician specializing in Noonan syndrome certainly would be able to tell. I can look at the cell images and see that there's a difference. So that's the idea of the project. They tested 3,000 variants in this initial pilot experiment. Now, as I said, we're scaling to 80,000, but not there yet. But in the initial 3,000, they found around 250 disease phenotypes that were discovered. Most of them were associated with pathogenic phenotypes. And here's some examples of what those phenotypes look like. And some are pretty common pathways that we noticed. So for example, it wasn't very common for there there to be a mislocalization between tubulin and cytoplasm, but it was extremely common to find mislocalization between the plasma membrane and the ER, for example. You can see a lot of traffic, let's say, coming between those two compartments. So here, again, we're trying to group these variants into classes where is it possible we could find a drug that would treat every single one of those maybe 30 to 50 that are in that pathway that are mislocalized between these two compartments? Maybe so. So let's give it a shot. How are we going to do this, though? Because at the Broad, we certainly can do drug screens, and we do a number of them each year, but they are expensive. And now we have 250 disease phenotypes. We can't afford to do 250 diseases times 30,000 or 100,000 drugs. So how are we going to break this bottleneck? We'll use technology that was developed by Paul Blaney's lab and then perfected by J.T. Neal in order to allow this kind of localization screening. And here's the basic idea, and which we're running right now in a pilot experiment funded by Ladders to Cures, where in each well, we have a single compound. So one potential drug already FDA approved. If it works well, it's like a really good candidate. So in each well, we put one single drug, but we put a mixture of the 250 or so diseases. And so it's the normal healthy version of the gene and also the unhealthy disease-associated version. Throw them all in there, but each one has a barcode, so we know which one's which. And that allows us to do this decoding, where the first step is we look at the localization. Does it have the healthy or unhealthy version of this localization? And then the second step, I won't describe the details, is where we do this barcode detection, where each little dot of light is telling us either G, A, T, C, and it's flashing over many cycles to tell us the identity of the barcode, which tells us which, for this little cluster of cells, which gene did they get, and is it the healthy version or the diseased version? So for each little island of cells, we find out, did this drug make the unhealthy version of the protein go back to its normal healthy location? And that's the idea of the project. We're just in the pilot stages of being able to do this drug screening. And so now we're hoping these 80,000 variants lead to many thousands of these phenotypes, which we can then put into this drug screening. Here we're testing it in a single cell type, but eventually we could test it over many over time. And so with that, I'm very happy to take questions and looking forward to the discussion for the rest of the day. Thank you. And please come right up to the microphone if you don't mind.
Thank you, great talk. Happy to learn so many exciting applications of imaging-based profiling. So my question is that, can you comment on the possibility using imaging-based profiling, how to design a combinatorial drug discovery?
How to design?
Combination drug discovery. Right?
Combination drug discovery.
Yes.
Yeah. So everything that I described, you can put whatever you like in the wells to treat the cells. So if you have combination therapies, if you think maybe 2 panels of drugs you might like to combine, you can absolutely test them together and see if they have the impact that you're looking for.
It's possible to use an AI-based algorithm to predict the combination.
Yeah. And so there's a lot of research out there to say, okay, if I perturb one gene, I get this output. What if I perturb 2 genes? Is it sort of the additive effect of the 2? Is it the multiplicative effect of the 2? Or do I expect them to interact in some weird way? So I've seen this more often with genes than with compounds. Like at least with genes, you have an idea of what you're perturbing in the cell. But the same principle applies for chemicals as well, where you can take large data sets with individual perturbations and maybe a small data set that has the combinations and try to learn from that to be able to predict the combinatorial effects. How well it works depends right now. I've only seen a couple of experiments in this, mostly in the transcriptomic space, not the image-based space. But it seems like it's at least promising, let's say. But I think it's too early to say whether it's going to be possible to really predict the outcomes from scratch like that.
Thank you.
Yeah, thank you for the question.
Hi, beautiful talk. So I'm thinking about how do you think about going from cells to patients, right? That discovery in the cells of changing the phenotypes. How do you consider all the physiology, all the pathophysiology, and all of that. What's your process of thinking through that?
Yeah, it's a very great point. So most of my research has focused on clearly the early drug discovery stage, right? You don't go from a cell experiment to giving humans a drug the next day. And so for most, I would expect that for most projects, if you did find an already FDA-approved drug that, let's say, looks like it reverses a localization phenotype in the cell-based assay, the next step in my mind would be to try to find some animal model of the disorder to try to see whether you have the same impact at the whole organism level. But it also depends on what the drug is. If it's a very innocuous, not too scary, vitamin or nutraceutical or something like this, you might be a little more inclined to give it a try. Whereas if it's a nasty cancer drug, you might be a little more reluctant to immediately try to give it to patients. It's beyond my pay grade. This is where the MD PhDs should get involved, but.
Well, I'm thinking of even ways of you're recruiting, large language models and other things to be able to inform better mechanism of saying, hey, this is more amenable to a change, right? Like something in a dish or something that's in a leukemia that you were describing. Those seem like just better targets, so to speak.
Yeah.
Is there some way of compressing the use right now of consultants and MD PhDs and actually having some more like logical flow that is programmed to evaluate these compounds you're generating?
Oh, it's a great point. So let's say, for example, out of an experiment, you get either a list of genes that look like promising targets, or you get a list of compounds or drugs that look like potentially useful. I absolutely see there's some potential for large language models that can synthesize the existing biomedical literature and just say, okay, out of these 10, these maybe make sense, or these might make me a little nervous because of these side effects, or so on. And so kind of interpreting those lists for clinicians to use their judgment as they're thinking about it, there's definitely potential there. But as everyone is well aware, there's also hallucinations. And so building these AI systems that are capable of accessing real data, providing you the direct link to the underlying data resource, those will be really important as we're trying to interpret these kinds of experiments.
Beautiful talk. As you may remember, I'm focused on ALS. I was yesterday at the Natural ALS Consortium to understand patients. 80% of patients who develop ALS we're calling, I guess, non-genetic, sporadic.
Yeah.
At the same time, when we take stem cells and generate motor neurons from these patients, it's kind of an undiscussed topic in the field, but they all seem to have phenotypes. And so for me, that says, and I think most people would agree with that, there's probably a genetic component even in what we don't call genetic ALS.
Yeah.
And so there's a lot of variants, obviously, and a lot of different genes and pathways that could be involved in development of disease. This is kind of a big bottleneck for the field at this moment because we don't understand which patients we should target with what type of therapies. Do you think, because I don't know how your variants were selected, but do you think your technology would also be able to kind of identify maybe unknown variants and their relationship to disease?
I love this question because especially at the Broad, we're pretty genome obsessed, but it turns out DNA is not everything and mRNA is not everything. Sometimes there are disease phenotypes that manifest at levels sort of beyond that, protein and onward, and just morphological infrastructural phenotypes. And so I think that profiling technologies like imaging and transcriptomics, proteomics can play a role in this etiology of like, what are these, how do we classify these patients into groups that make mechanistic sense and might be treated in the same way. And so a whole nother approach I didn't describe today is to take patient cell lines and grow cells in a dish. We've done this for a few disorders so far, where we try to identify from the patient samples, are we able to group the samples in some kind of meaningful way that gives us some kind of clue as to what the shared mechanisms might be in different classes of patients. And I think that's a real potential here.
And have you thought about taking variants from patients and introducing those variants into cells?
And then making isogenic cell lines. It turns out if you want to make an isogenic cell line by introducing a mutation, it's a very clean experiment from a design perspective. It's a little messy when it turns out, because when you clone the cells, they turn into multiple independent snowflakes. And so if you don't even knock out the cell, but just knock out the gene, but make 10 copies of a cell line, they all look a little different from each other. So if there's a strong phenotype, you will certainly see it in the isogenic case, but it's not quite as easy and simple as one might hope.
So if phenotypes are weak, it's still very challenging.
It's still challenging, but the only way to know is to try. So empirically, I think it's a really good thing to try.
Okay, thank you.
Yeah, sure thing.
Hi, thank you so much. It was a really wonderful talk. Question was already commented on a little bit, but I was curious about the technology and its application in understanding variants of unknown significance.
Yes.
If you could look at using the different localization to try and understand those better and maybe reclassify.
Absolutely, that's really the fundamental goal of that project is to for the pathogenic ones to keep moving along towards drugs, but for the unknown significance ones to group them. Do these look more like the pathogenic or more like the benign? So that is absolutely one use of this kind of data, and we've seen a number of different examples. Sometimes it's like pathogenic and benign. Other times it's like 3 different classes of pathogenic and then benign. And sometimes you can't tell the difference at all because it's not a localization problem, it's some kind of other functional problem. So we see across the board all different things.
Thank you.
Okay, thanks everybody.
That was amazing. But following up on our long, now 10-year history of celebrating this particular day, we've always been very focused on, in equal parts, celebrating the science and actually the other members of the community who are just as important, either because they're scientists themselves or because they're pushing the science forward. And of course, I mean the parents or the family members, the advocates, the people who really make this community work, I think. And to paraphrase Margaret Mead, do not underestimate what a motivated group of parents can do to change the world. Indeed, it's the only thing that ever has. And so on that theme, it is my honor to introduce you to 2 parents who are doing incredible things in this area. So Ada Leo and Nathan Guo are both here to represent the ZTTK Sunshine Foundation. I think they'll tell you much more about their work, but I just want to say for our Ladder Cures Accelerator, an important collaborator is also the Chan Zuckerberg Initiative, and they have been successful in securing funding for their organization. And so it's wonderful to explore ways in which we can all work together, the scientists and the parents and patient advocates. So I leave it to you to tell us more about your work. Thank you.
Thank you so much. Thank you so much to the Termeer Foundation and the Broad Institute for having us here today to speak to all of you. My name is Ada Leo. This is my husband, Nathan Guo. We are parents to our 2-year-old son, Lucas, whom we call endearingly Luki. We're also patient advocacy group leaders of the ZTTK Sunshine Foundation, advocating for Luki and others like him with ZTTK syndrome, an ultra-rare neurodevelopmental disorder. Today, it is such an honor to share our journey to accelerate a cure for ZTTK. I will start off by first defining what is even a cure for us and how critical it is to integrate patient insight in our journey. And then my husband Nathan will walk through the research roadmap integrating patient insight and how some of you can share your innovative work as part of our journey. We hope you will walk away today from our talk with more insights into how partnering with patient advocacy groups like ours can increase the impact in innovation in rare disease research. So first, I want to get some insights from the audience. What is a cure? How many of you here think you have a precise definition of what is a cure by a quick show of hands? Great, I see a few hands Now, by another quick show of hands, how many of you are quite uncertain about what is a cure? Great, I see many hands in the audience. Great. In fact, when we talk to other patient advocacy groups, and even within our own ZTTK community, there is no unified definition of what is a cure. So how do Nathan and I think about what is a cure? We would like to borrow a framework from healthcare professionals. This is a framework they use when administering safe medications. We believe it is also applicable to how we think about rare disease research. It is giving the right drug at the right dose, at the right route, to the right patient, at the right time. While we think dose and route are important, today we're going to focus on defining the right drug for the right patient at the right time. To understand what is the right drug, we have to first understand what is the disease. ZTTK syndrome was discovered in 2016 by 4 researchers. It is an ultra-rare neurodevelopmental disorder. It is a monogenic disease caused by a mutation in the SON gene. It is diagnosed primarily through whole exome or genome sequencing. Our community is growing. We have estimated roughly 300 families around the world. We believe many remain undiagnosed today. Our phenotypes and symptoms are a wide spectrum. What we know today is that the most prevalent symptoms are global development delays, motor delay, speech delay, feeding difficulties, and also intellectual disability. Based on a paper published in 2021, we know that from the initial ZTTK cohort, about 50% of the patients develop seizures during childhood. There is currently no cures or treatment. So by looking at this, one might think that one important concern would be to address the seizure burden. We know that some other neurodevelopmental groups have also identified seizures as their primary endpoint and even have drugs in development. And this is where patient insights are critical. Last year, we conducted a survey with our ZTTK community. We asked them, what are the top 3 symptoms that are most important for you and your family to address in your child? Cognition, speech. Seizures come in lower than these other symptoms. We dug a little deeper to understand why. We collected stories, anecdotes from our families. Some mentioned that they prioritize cognition because they hope for their child to have independence one day. Some say that they prioritize speech because they would like their child to express themselves more freely, even if they're just basic needs and emotions. And this is where we believe patient voices are so critical for us to define what is the right drug. But even when we have what matters to the entire community, each family has their own unique journey. They have their own dreams and hope for their children. It is up to the individual patient and family to define what is the right drug for them. I can only speak for what matters to me, Nathan, and Luki. This is our 2-year-old son, Luki. He is sweet, curious, and he is clever in his own way. He's also experiencing profound challenges. At 2.5, he spent hours every single day working on the most basic skills. The first picture is of him working on standing and walking. He's in this big equipment called a stander that he has to be in for at least an hour a day to help with standing. Initially, it would break my heart to strap him to this big equipment, and he's strong. He would fight me, kicking and shouting to get out of the stander, but now we are more used to it, and we even have some fun. This is actually a photo from last year, our photo shoot from Rare Disease Day. The second picture shows that he's still working on his fine motor skills, how to point, how to pick up a cereal. This is from a 3-week intensive that we were in last year, where he was in therapies 3 hours every day for 3 weeks, OT, PT, speech therapies, just to work on these basic skills. In his picture, he's working on crawling, and after the therapy sessions, he was able to put a ball in the container, and he was so proud of himself. The look on his face was priceless. Now, Luki is desperate to communicate with us. The only word he can say right now is mama. He says it constantly. He says it to everyone and everything. But I cherish it so much because I've also heard from other community members that kids may lose their speech. The few words that they have acquired, there may be regression over time. The other picture is of him drinking from a straw cup. Every meal, he's working so hard on basic skills to chew, to swallow, and this is one of the first pictures of him being able to drink water out of a straw cup. Today, he's still primarily fed by a feeding tube. Fortunately, he has not had a seizure yet, but it is a fear that is looming over us every single day. While we don't know when Luki will be able to walk, to talk, to feed independently, what we do know is that with each day that passes, his brain is not developing the way it should be. We love Luki for who he is, but we also want him to fulfill his potential beyond the limitations of ZTTK. The right drug is a drug that can help him right now. The right drug may not be the drug that can help him reverse all his symptoms. The right drug may be a drug that can improve his cognition, his speech, even just a little bit. Because like some families in our community, we also hope for him to have independence one day when we're no longer around. We too hope that he can express himself freely, maybe one day say, Mama and Papa, I love you. As we sit here today discussing groundbreaking research and innovation, I want to thank all of you and encourage you to think beyond the molecules and data. You are giving an opportunity to children like Luki, an opportunity to rewrite their future. Thank you so much.
Great, so before I start to dive in further into the science of this all, I want to first acknowledge that Ada and I are very lucky to be here. Today, drug development for ultra-rare conditions is largely driven by parents which is very empowering, but we have to acknowledge that only parents that have sufficient resources, the ability to access networks like the Broad, are able to really be successful at this. And so we feel a deep obligation to our community, some of whom are just trying to survive the day-to-day, many of whom do not have the resources to be able to progress these treatments and cures. So it's really important to acknowledge that first up front. So in the next few slides, I'll take you through more of the science of this, right? When we think about the right drug for the right patient, of course we need to understand the underlying biology and then put that together with the right time. What does that mean? So on this slide, you'll see a lot of detail about ZTTK and the SON gene biology. It's a heterozygous mutation. It's a haploinsufficiency and loss of function. We haven't seen any toxic gain of function. Almost all of the mutations we've seen in our community are de novo. You'll see to the left side here, both the SON gene and the SON protein. You might see a very large exon 3. You might see both DNA and RNA binding motifs. You'll see a lot of little triangles along both the gene and the protein. Each triangle represents one patient documented by our lead researcher, Dr. Aaron Ahn at the University of Alabama at Birmingham. What you'll see is that scattered across the bottom, there's a lot of 1Z, 2Z mutations, and then there's also one big spike. That spike is actually about a quarter of the population has the same 4 base pair deletion, resulting in a frameshift mutation. We're still understanding the genotype-phenotype correlations, but we're continuing to learn more about the SON gene and its function. Our understanding today is that the SON gene is quite important. It's a nuclear speckled protein. It's believed to be a master regulator of many downstream genes, actually many of which also have their own intellectual disability disorders attached to them. It's important for intron excision in pre-mRNA splicing, and it's also critical to some regulation of some transcription pathways. And it's highly conserved across species, and it's expressed ubiquitously in the body. So now that we understand a little bit more about the disease and the biology, we really want to present kind of a framework of how do we prioritize our ZTTK drug development efforts, because as a patient advocacy group, we have limited resources, time, and money to be able to pursue treatments and cures for our kids. So what I'll show on the following page is a 2x2. Both Ada and I are ex-consultants, so we think everything can be solved by a 2x2. On the bottom x-axis, what you'll see is speed to the clinic. Speed to the clinic is really the time dimension of this. Are there existing approved or clinical stage drugs for CNS, ideally even for rare or ultra-rare CNS, that can indicate that there are real regulatory pathways we can leverage, right? It's much more difficult for us as an ultra-rare group to try totally brand new pathways. And then on the y-axis, what you'll see is kind of the potential therapeutic impact, really the right drug for the right patient, right? Does it actually address the patient need that Ada talked about earlier, that the community has identified, that the patients identify as their primary need? What's the mechanism of action? How safe is it? And ultimately, what's the patient experience with that? So here's the 2x2. I'll walk you through the different pieces, really a 3x3. So as I mentioned, on the x-axis, speed to the clinic. So on the far right side, that is the fastest to the clinic, things that are much more likely to be proven. On the y-axis, what you'll see is kind of the potential therapeutic impact. And for those of you who are astute, you'll probably also already figure out that you ideally want things in the top right corner, right? Things that are fast to the clinic and have high therapeutic impact. Unfortunately, in our case, this one's a little empty. Our protein is quite large. It's not able to fit in an AAV9 vector. Also, we have not yet identified any obvious ASO targets. So as a result, that remains empty for now. As we move into the blue category, these are the ones that we think have the most promise. But today, what we're doing is really spreading our bets across these to understand what might bear fruit. And then finally, you'll see also a number of innovations that have come out of the Broad, a lot of advanced modalities that are just entering the clinic, still in early translational research, but maybe too far in the future for us to seriously consider at this point, but obviously hoping that those may accelerate. So a few comments about this overall framework. First, this is really disease-specific and even to some extent mutation-specific as well. So for example, in our case, it's a 4-base pair frame shift mutation. That has different drugs applicable to it than, say, an in-frame deletion or a whole gene deletion. So the context here is really important, and you could really draw a different 3x3 for every disease and even subclasses within the disease. The other thing to point out is this is not a static chart. This is just a view of what we know today, and really the goal of making bets in all of the blue category here is to identify, are there pathways that might move that into the top right corner? These modalities will eventually become actual hits and then actual leads, and that will be what progresses over time, but this is what we really hope to kind of arm our own efforts and other patient advocacy groups as well. So maybe just to take you through a few examples. So gene replacement therapy is a really common one. I mentioned earlier AAV9 single vector gene therapy ruled out. You'll see in the middle the truncated AAV9 gene therapy, so a mini SON construct or something like that. Today, our assumption is it may not have the full efficacy, but we just need to do more research to understand how much do we actually need to boost SON gene function. And then you also see dual vector gene therapy in the top left. Obviously a lot of experimentation, but no kind of clinical dual vector strategies for CNS. There's a couple for ophthalmological diseases, but not for CNS. Another example is ASO. Exon skipping ASOs kind of out of the picture. You saw that very large exon 3. We can't really skip that. But we are looking at ASOs to promote full-length SON expression by kind of inhibiting other isoforms, and we'll continue to explore other ASO pathways as well. Finally, you'll also see for the gene editing technologies, many of these have come out of the Broad, of course, but in our case, CRISPR editing and base editing don't quite work for a frameshift mutation. Prime editing may be a good candidate, just given we really just need to knock in 4 base pairs or just get it in frame. But hopefully this gives you a bit of an understanding of how we might think about, on a disease-by-disease basis, how to tackle the problem at a systemic level. And finally, I'd say all of these kind of different modalities are interdependent and context-dependent. So in our case, actually, as you've seen, we've walked through a few of the other options from a genetic medicines perspective. In our case, drug repurposing may be the most promising route because some of the other ones have been ruled out, and especially from the speed to the clinic perspective, this may be by far the fastest option. So really, I would encourage the audience to think about when you're building up the picture of how to leverage innovation to really have the impact that we're all hoping for, we really need to think about the holistic picture of the right drug and the right patient at the right time. So, today, the way we're approaching this is we're really operating a preclinical biotech out of our foundation. So just like any other preclinical biotech, what we're doing is understanding the natural history of the disease, which is very limited given that it was just discovered in 2016. Our lead researcher, Dr. Arya Rao, who I mentioned earlier, published on the first ZTTK mouse model last year. Some of my colleagues at Thermo Fisher developed the first ZTTK iPSCs, and we're working on quite a number of projects to identify both targets and hits, and also to verify those, including some projects with the Broad as well. So on this page, you'll see we have a lot of projects going on in this next year. I'm not going to go through all of them, but what I would highlight is, there are projects that we're still looking for partners to work with us on. So specifically, proteomics to understand which SON isoforms show up in which organs, to understand kind of what do we need to focus on in the CNS versus liver versus stomach, et cetera. Also, trying to better understand what are the critical epitopes to ultimately inform SON antibody and ELISA assay development. And after Anne's fabulous talk earlier, I think we also need to add a cell morphology project on here as well. So, I would really encourage you and the audience to think about it, the framework here that we've presented, the right cure for the right patient at the right time. And I think for scientists at the Broad and other research institutions like this, no one wants to do innovation for innovation's sake. People want to do innovation to drive real impact. And I'd say that the best way to realize that impact is to partner with patient advocacy groups like ours, parents like ourselves who are learning the science and could use your help and partnership in accelerating the work. I'd like to thank quite a number of people, our support network, Dr. Erin Ahn, our lead researcher at the University of Alabama at Birmingham. She is also a board member of our foundation and was a co-discoverer of ZTTK syndrome in 2016. We have our foundation, of course, our scientific and clinical advisory boards, and many, many partners who have shared in-kind resources support all along the way. So if any of you in the audience are interested in supporting Ada and I on our journey to cure ZTTK, please feel free to email us at nguo or aleo@zttk.org. And with that, we'll open it up to questions. Thank you.
I don't need a microphone. I'm surprised you're not mentioning the one generic approach, which is to enhance splicing of the wild-type copy of the gene. That has proven very effective in spinal muscular atrophy. There are now clinical trials for a haploinsufficient genetic disorder of the central nervous system, Dravet syndrome. It appears to be a genetic type of therapy where what one needs to do, and Tim, you on your advisory board knows how to do it,
Yeah.
Which is scan oligonucleotides to simply find those that will enhance splicing of key exons in the wild type copy of the gene.
Yeah.
And that could be a generic therapy for many haploinsufficient genetic disorders.
Yeah, absolutely. I'm not as deep on the oligoscience as Tim U. The U Lab did spend quite some time scanning for kind of obvious ASO targets, and unfortunately they didn't come up with anything. I know that, of course, Stoke Therapeutics is working on the Tango approach. We've had some initial conversations with them about it, and unfortunately it doesn't seem like it's applicable at this time. But continuing to look for other options with that strategy in mind.
Computer is not necessarily the best approach.
Thank you.
Thank you.
We'll take note.
Thank you for your talk to sharing your personal moving and inspiring stories and a strategic roadmap for drug development. So my question is that, of course, the cell and the gene therapy offer a final cure promising for the disease. How do you evaluate the possibility we are using the nutritional interventions, like food supplementary, to intervene the progression of disease and ameliorate the symptoms of patients?
Yeah, it's a great question. I think part of what we're working on whenever we're thinking about gathering more natural history is even just what are patients taking today? Even just gathering the data on what medications, what supplements, what diet, et cetera, have patients been using might unlock, hey, actually there's quite a few patients that have converged on the same kind of therapies, whether just food or medicine or otherwise. Those might even evolve over time. It might be different in childhood versus adulthood, et cetera. So It's something we're working on to really gather that data. Actually, one of our partners, Citizen Health, is really helpful in aggregating a lot of the EHR data to be able to just observe, at least historically, what is our community doing. But it's a big focus of ours. We're also working on a project with Combined Brain, which is another nonprofit in the neurodevelopmental space, specifically to do metabolomics of human patients to understand, are there differences? We're still trying to disentangle, are the neurological features of the disease actually being driven potentially by metabolism itself? So we're continuing to investigate that as well.
Thank you very much.
Thank you.
Good afternoon. I'm Catherine Smith. I'm the executive director of the Termeer Foundation. Apologies, I've sort of lost my voice today, so we're gonna muck through this. It's gonna be short. First of all, I just wanted to say to Nathan and Ada, I'm not sure where you guys went, but thank you for introducing us to Luki. I've been following the progress of the work that you've been doing, and it's tremendous and impressive, and thank you for all that you're doing. I'm going to be introducing Richard Novak. You can come on up if you'd like. Richard is one of our 2024 Termeer Fellows, and he is the CEO of Unravel, and you would have seen probably their logo up on the last presentation. And Richard's going to be sharing with us about the partnerships that he's done with patients and share more about what that looks like from the first-time founder, first-time CEO side of things. So welcome, Richard.
Thanks so much. I think I'm mic'd up. Yes. No, thank you so much. It's an honor to be here. It really is. Thank you to the Broad, to Anna, to the Termeer Foundation for this wonderful support. As a relatively newly minted member of biopharma, I'm here to share both good news and bad news. Bad news, biopharma really stinks at making drugs. We get it done eventually with enough money, I think, as Anna pointed out, but we're really terrible at it. The good news is we believe there's a really good solution, and that's in the patients themselves. They are part of the problem that we're trying to solve, but they're part of the solution. And so what I'm here to present today is not so much the science, though we're going to dive into it a little bit. It's really how do we make this infrastructure work for everybody? And so our strategy at Unravel is patients first. That is the problem. That is the North Star. We're singularly aimed at that, and let's work together to figure out how we get there. And we're doing this through a very heavy partnering approach. So here is traditional drug development in a very abstract sense, but you can see that there's a target. This usually comes out of academia. Mouse highlights any sort of preclinical models, cells, animal models, et cetera. And then you get into the critical question, does it work in patients? This is what we've been trying to do this whole time. And this is where my earlier bad news comes in. We're terrible at this. It takes years, if it's even going to be successful. On average, a new target has about a 98% failure rate in terms of this approval process. So this doesn't work. And this is for non-rare diseases too. But when you get to rare and especially ultra rare, this just doesn't work, right? So what can we do? And what, kind of digging one layer deeper, what's the problem? Our thesis is that people are trying to treat disease, when really we should be thinking about treating patients. Right? Diseases are not the same as patients, and so a shout out to the Cleveland Clinic for these really helpful graphics, but they represent the generalities. They don't represent individual patients. So again, what we say is it's the patients, not the disease, that responds to the treatment. And here's actually a picture from our office that reminds us this is why we're here. And actually, Luki is on there as well. And so, this is not a new concept. I would love to be able to take credit for it, but it actually goes back to the 19th century with William Osler, so the father of modern medicine. Basically, the good physician treats the disease. The great physician treats the patient with the disease. And I think we've lost track of that along the way, and so we're trying to bring that back. And part of it is because the technologies today enable it, and part of it is because the patient advocacy groups are so freakishly sophisticated and motivated that now we can do it, right? So it's this kind of, it takes a village, but even more complex concept. And so what we did at Unravel is we have this patient-driven drug development. And there's, people pay lip service to patients, and every pharma company has a page that says, oh yeah, we're patient-centric. That's great. But have you actually worked with these groups? Have you worked with the patients, right? And that's sort of the question. And so, again, why do we start with patients, right? And to some extent it's obvious, but to some extent there's really deep biology. And the scientific problem is a really big one. And I think Anna mentioned this really nicely in the beginning. We have no clue what half of the pathways are doing. You have these proteins, genes that have been identified. That's great. No idea what they do, or maybe we know one thing out of 100. The reality is that what constitutes a patient, right? Now, not just a human, but just a patient specifically. Sure, genetics in the case of genetic disorders, the environment. This includes diet, medications, et cetera, and time. And I think Ada and Nathan really nicely alluded to it. When is a really critical question to ask, not just what and how and who. And so what we did, and we spun out of the Harvard ecosystem, so just across the water here, out of the Wyss Institute, and there we had this insight that these complex biological networks drive clinical function. And here's actually a real example of one of our networks, and the idea is that these interactions, as opposed to any one component of this, drive patient outcomes, right? It's kind of the sum of all the parts. Now, if you have tried to drive through Boston, for example, just coming here today, there was a malfunctioning traffic light. Turns out that backed up the traffic onto Sturo, which backed up onto I-93, which who knows what it's doing, right? So one traffic light can have these vast effects. That's a network problem that you encounter every day, especially in Boston. And so let me walk you through how we do this. So obviously this is teeing up to a computational example, as you might imagine, but the idea is that you have this healthy network, and again, just completely arbitrary. This is actually from a Rett syndrome patient I'm going to talk about later, but don't worry, the yellow lines are just for show here. You have some sort of healthy interaction. This is what normally should be happening. And there's a problem. There's a gene maybe that's mutated. There might be an infection that's causing something. Who knows what it is? In this case, it's actually the MECP2 gene that's highlighted here. There's some problem. There's a mutated node, and you heard this nodal biology is really kind of getting at that. So great, it causes some disruption. It's that malfunctioning traffic light, right? So how do we fix it? Well, you could fix the traffic light, but you can also fix anything else that rebalances this network. And that's the really great opportunity in biology. It's a mess. And so why focus on just one thing, right? You can take any of that mess and use it to your advantage. As unfortunately every oncologist knows, cancer cells are really good at finding these alternative pathways. So why can't we use that for good? And that's really what we do at Unravel. And essentially what we do, we're forecasting how patients respond to therapies. Again, it's about the patient, not the disease. So we have patient RNA signatures from actual individual patients that we work with on a daily basis. And we have drug RNA signatures, and not unlike what Anna showed with the visual signatures, these are really transcriptomic signatures, but from the perspective of these networks. And then we go through this magic, and we ask, will it help this patient, right? Lukey, for example. Can we treat all the patients within a particular disease, or however you want to look at it? The answer may be no, but you can treat some of them in one way, and you have to find something else for the others. Are there better treatments? So repurposing might be a great place to start. We agree, but we're also developing new drugs that are now tailored to the mechanisms and targets that we discover. And one thing I want to mention is that also looking at the adverse side of it, right? So you make all these molecules. One of the biggest drivers of costs are all the failures, toxicities, lack of efficacy, and all that. But toxicities are, put patients at risk and are just bad business. So just, there's no reason to not avoid that if you can. And so we see all of that in a pretty clear way. So we bring this function-first approach to drug discovery, and we have these three components. One is BioNav, and it's really this Google Maps for health, this network biology of human health, coupled with an in silico compound library of about 40,000 molecules. Now, all these are purchasable. You could say this is repurposing. To us, they're discovery tools. And over time, families and foundations like the ZTTK Sunshine Foundation came to us and asked, well, can we just use those tools for the ones that are actually approved for patients? And the answer is yeah. We're not a repurposing company, but we're trying to probe the complexities using these molecules. One thing that we do as an intermediate step, we developed this concept of squishyware. It's not hard, it's not soft, it's squishy. These are tadpoles. They are actually very squishy. And we can do sort of a kind of computer vision type analysis, like again with with the cells that you saw earlier, but with living, moving tadpoles that are, again, mutated with the various genes. And we can just ask, does it help rescue that model? But then really where we're at, again, it's patient-driven and patient-focused drug development or the clinical studies. Does it actually work in a patient? And so what we do, we use existing drugs to ask that question because you could administer those today, not in 10 years, right? With potential other toxicities and other things. Yes, they may not be ideal. They may not get you the 100% of the efficacy you're after, but if you can get 50%, one, it proves out your mechanism, that you can now invest those resources for a new drug, and it might help patients today in the meantime, right? So you don't have to wait 10 years for the perfect drug and have nothing. You might have an intermediate step in between. And so does it work? Well, yes, it works, even to our surprise, quite well. We've actually shown this actually during the lovely pandemic that we had a few years ago. We showed that we can predict the function of specific statins in this case, so used for cholesterol control. You predict specific statin impact on survival from COVID. So these are patients who are getting statins because of their hyperlipidemia anyway, and we showed that specific molecules, but not the class as a whole, were actually therapeutic. And this was death as an endpoint, so pretty cut and dry in terms of outcomes. And we were statistically significant. We had a false negative, which is a good one to have, but overall much better than your 2% success rate. We had an 80% success rate in predicting clinical outcomes. And so part of the challenge here is really this infrastructure and how do you work with patient groups, how do you partner? And I mentioned that families started coming to us, and so we actually had to develop a new business model. And we set aside the one that we started out with about 4 years ago before spinning out. And we said, we really need to offer these services, even though we're not a service company and we're not a repurposing company. And so we started the RareShift service platform. And really, this is just sort of cost-based drug discovery for nonprofits. It's rareshift.org. And there it is drug repurposing. That is the goal. That is the message. That is the goal. For us, it's a stepping stone towards the novel drug arm. Essentially, it provides clinical discovery and validation. And then the partnerships build in the process. And we become a partner to the foundations or the families, and they become our partners in the discovery and validation process. So it becomes a win-win. And what we've been able to do is actually using nasal swabs. So again, before the pandemic, this would not have been possible, to collect a few cells on a COVID test kit, essentially, and use that to extract RNA, run it through our BioNav platform, and get insights on the brain, because most of what we work on are neurological diseases. And then we can use this AI platform to figure out how do we potentially fix this patient, what's going on, how is patient 1 different or the same as patient 2, and so on. And what's really exciting, we can work all around the world. So we've actually sampled patients from the remote outback. Took 5 weeks, but we did it. We can basically ship these kits all around the world at room temperature. And so what we have started to do is see the evolution of these diseases into truly what we call therapeutic response groups. It's not about how you got there, it's how you fix things, right? And so that's where these TRGs, therapeutic response groups, come in. And so this is actually SYNGAP1 is another one of these neurological disorders. If you take a look at this rainbow matrix, there is no pattern. We tried. There's nothing really clear. Different ages, different mutation sites, genotype, phenotype doesn't really exist. But what we start seeing in terms of drug response are 3 groups. And they actually correlate really beautifully. And those patients are actually quite different from each other once you look at them from that perspective. And so now we're trying to take these really complex populations and reduce them to something more manageable. And so just to show what we've been able to do on the biopharma side of our company, we work on Rett syndrome. It's, again, one of these neurological neurogenetic disorders, one of the more common ones. This is not rare, and there's been one approved drug on the market. And we actually use that as a benchmark, and we basically did the same process, collect data from patients and from other sources, run it through our platform. Here's actually the mutation, but we found a target somewhere else. Doesn't matter what caused it, but if you can engage this target right there, right? How we got there is the magic, but we show that if you can actually engage the target right there, it fixes the network. And that was the real insight. And so now we actually have a new drug for this, but we're not going to develop it until we show that this works with an existing drug, because that's how we can test it today. By the way, the existing drug, Verinostat, formulation in Unravel 1, outperforms the one that's currently on the market dramatically, especially in the later stages of the disease. And we're actually starting 2 clinical trials in Rett syndrome. And then you may ask, does it actually work? Well, this is the beauty of using existing drugs. There's actually a family with a boy with Rett syndrome. So boys are really rare. They tend to not do very well with this disease. And they were really looking for options. Their clinician was looking for options. And so long story short, we identified vernistat actually to work for that patient computationally. And the clinician felt that it was appropriate for her to prescribe it to the patient. And sure enough, extremely low doses. And this is a cancer drug, right? So keep in mind, this is not a supplement. The sleep quality improved pretty dramatically. Normalized blood metrics. This is an objective measure of improvement. And then improved motor and cognitive function as assessed by various clinical professionals. And now we're actually, the boy's still on it. This has been on it for over a year at this point, continues to improve, and we're actually layering additional therapies on top of that. I mean, the clinician is, but with our data. And what we're doing now, this was really exciting to us, because saying, look, this really can help even reverse this extremely severe case. We're actually starting 2 clinical studies, one in Rett syndrome in Colombia. So I joke that we're bringing white powder to Colombia for a change. It's a really fantastic place. We go to where the patients are. There are 15 patients all being treated by a single clinician, single site study, really fantastic place. And they have never been able to participate in cutting edge research like this. And so this is what we're doing. We're actually using oral liquid vernistat, validating our target, right? Now there's a repurposed candidate. We have orphan drug designation for it in the US. We will bring it to the US. We also continue with it in Latin America. But then we're actually building Ravel 2, which is this new optimized drug specifically for Rett syndrome and several other diseases. But we ask the critical question, does it work before we commit tens of millions of dollars to developing a new drug? And it gets even better. So the question is, there's 15,000 rare diseases, or whatever the number is, it's a really big number. How do we tackle not one at a time, but how do we parallelize it? And so we've actually identified these therapeutic response groups to span diagnoses. So there's Pitt-Hopkins syndrome, which is not Rett syndrome. It's a different gene, different disease, with some similarities. But they're actually sponsoring a clinical study because we predicted that it would actually be quite effective, predicted that it would work in mice. Lo and behold, it worked in mice. So now we have to go to patients. That's the only way to answer that question. And overall, we have about 50 different diseases in our portfolio. As you can see, they usually start with this BioNav screen. Some of them have tadpole models that get developed. Some don't. Some have mouse models that get developed. Some don't. We work with Jackson Labs as well. Where people are really headed is the clinical part, right? Does it work in actual patients? I mean, that's the ultimate question. And so all these are off-label. I should note that we do not treat patients, but we generate research use data that can be used for clinicians to evaluate as they see fit. One thing I want to announce here today, so we actually have not announced this yet, so this is the first time. We just signed a very key agreement with Combined Brain. So Nathan and Ada mentioned them. They're an umbrella organization that really spans about 120 different patient advocacy groups, encompassing a little bit more diseases than that. Some of them have actually more than one falling under them. And essentially what we're doing is taking our really patient-first approach, RNA-driven approach, and applying it to not just the 50 disorders that we already work with, but another 120, right? So this represents really a landmark partnering in the space. Rare disease, most of these are ultra-rare diseases, ultra rare. I mean, one group has 6 patients documented, right? So these are, talk about a challenge of scaling to commercial opportunities. And this is the type of partnering that I think we envision, that we, thanks to all these fantastic patient advocacy groups, are really trying to change how we do drug development. So with that, I want to thank you for your time, and again, drug development driven by patients.
Thank you, wonderful talk. So you mentioned the powerful pipeline of BioNail for clinical usage. So can you comment the current practice in regulations of FDA? How can you to meet with FDA regulations? And you talk about the clinical trial in Colombia. It's a possible solution to navigate the FDA regulations.
Yep. So the regulatory aspects are definitely key. Again, it's a very, right now, the last month or so has been very dynamic in that space, so we'll see how it plays out, but I'm sure it will. I'm sure it'll stabilize. So that caveat aside, absolutely key to engage with FDA early and often. We filed a pre-IND in the US before we went to Colombia. We want to make sure the FDA agreed generally, not necessarily to some of the specific details that are more country or regulator specific, but would this make sense, right? Before we go to where it's faster, cheaper, easier to do a trial. The regulation actually in Colombia is very similar to the US, but the patients are there. The cost of doing the trials is just a fraction of that in the US, as you might imagine, right? So absolutely key to engage with the FDA. BioNav, we have no vision for that to be a clinical tool. To us, it's a discovery tool because the regulatory challenges dealing with these networks, I don't think are possible to overcome, just given how the FDA regulates diagnostics. That said, what comes out of it is a drug. We can regulate the drugs, and we can be absolutely compliant with that and do so in a very efficient way. That's also where repurposed drugs have an advantage because a lot of the safety, the main safety concerns are addressed. Now the question is it safe in this population? But you sort of know what to look for. Is it kidney toxicity, liver toxicity? You have a better idea of what to look for. So it tends to accelerate the whole process and reduce costs. But the regulatory aspect is really key to bring in early. Yeah.
Thank you.
Yeah, thank you.
Hi, I think it's amazing that you're able to actually develop therapeutics for so many different diseases. But how do you actually, on the actual biotech company, prioritize which diseases to actually pursue the clinical trials for?
Yeah, that's a great question. This is where being in rare actually has its advantages. So orphan drug designation gives you priority review, it reduces the time for review, it eliminates a lot of the fees that pharma typically has to pay. So you have all those benefits to to advocate for it. Now, which one of these rare diseases within the rare space, this is where it's a partnership, right? And as with any partnership, some are way more dynamic and fast, and others are more slow, and there are different priorities that the different patient advocacy groups have that may or may not directly align with the given trajectory at any one time. And so it's a sort of group decision of like, what makes the most sense? And it may be that you pick one, for example, with Rett syndrome. I think we're more likely to get investor interest for a US trial for Rett syndrome versus one of the subgroups that we found already has 40 patients globally. Probably not a good thing for investors for a pitch. That said, they may be a really good opportunity for getting EU approval for that drug. Because once the drug is approved in a given country, you can start selling it. And clinicians can start prescribing it off-label, right? And so, especially for Ravel 2, that new drug could go through this 40-patient group much faster in a 5-person study. And so like the commercial and the approval side are not always as aligned as they are for large diseases.
I figured it'd be
So,
complex to stop.
yeah, yeah.
Actually, just to pick up on this theme, have you considered doing basket trials? Is that something that
Absolutely.
FDA would be open to?
Absolutely. So we like it. Talk to your neighborhood regulator about it. In fact, in Colombia, we propose that they do not, on principle, allow basket trials. So we're doing 2 separate INDs, 2 separate everything, even though it's more copy and paste for all the documents. We'll do that at least at the beginning. And hopefully that'll evolve as these things change. And this will also be a factor of the patient advocacy groups advocating, hey, there's no way we can do our own trial, but can we have 3 patients in this trial, right? So I think that will be kind of two-way street there. That said, in the US, the US is open to that, the US FDA. It has its own set of challenges, but absolutely there's lots of precedent. Yeah. Great.
Thank you.
Thank you.
That was remarkable. Basket trials, remember that, because all of our efforts in nodal biology will ultimately be served by, being able to use the same medication for patients with multiple different genetic misspellings that seem to converge on a biological node that can be targeted. That's exactly what we hope to accomplish. So this is an important point that was made. So I'm glad that it was made. It's been successful in cancer, and the US FDA is open to it. So we have to all advocate as a community for this structure to be utilized for rare diseases, and to also provide examples where that can happen. Speaking of the future and, people working within the context of the Ladders to Cures Accelerator and the broader Broad community toward that incredible future, and maybe also building on Harvey Lodish's incredible suggestion about identifying ways in which you can increase the activity of certain genes and how you might be able to systematically do that. I'm wildly proud to introduce Arya Rao, who is an MD PhD student here at Broad, working in the laboratories of two incredible physician scientists and colleagues, Pardis Sabeti and Sangeeta Bhatia. And she's going to tell you about her work harnessing evolution to try to understand how we can regulate genes. So Arya, take it away.
Thank you so much, Anna. And it's great to be here with you all. This is such an amazing community, and I'm fortunate to be a part of it. So thank you to the Termeer Foundation, and a big shout out to Jillian for all of her help today and in general. I'm excited today to share with you in particular my vision for the next generation of rare disease therapeutics and how we can achieve it with insights from evolutionary biology. Back to basics, our genetic code is made up of 4 simple letters, A, C, T, G. So with different combinations of these bases, our genomes are able to execute this huge number of functions necessary to make our bodies work. This is the language that evolution has used to sculpt our intricate human physiology. But it turns out that evolution has a really narrow vocabulary. The human genome is 3 billion base pairs long. And so if we do the math, there are 4 possible bases that we can have at each of those 3 billion positions, which means this. For a genome of our size, there are 4 to the 3 billionth possible configurations. That's more than the number of atoms in the observable universe. But less than 1% of these have been explored by natural evolution. So what's going on with all of that missing code? Evolution works with 2 main forces, mutation and selection. As our cells divide throughout our lifetimes, our genomes are copied over. Errors are sometimes made in this process called mutations. Mutations that are beneficial are selected for. These rise to high frequency and then are passed on from generation to generation. We can imagine evolution like a hiker who's going through this big mountain range. Each step that the hiker takes is a mutation. A beneficial mutation is a step forward, closer to the top of the mountain, whereas a harmful mutation is a step back down the slope. Here's the problem, though. Our hiker, from where they're standing in the mountain range, does not know whether the path that they're on is the path that will take them to the tallest peak. And so it's easy for them to get stuck on a suboptimal peak because from where they're standing, they don't know what's up, what's down, where to go. It's for this reason that evolution has only explored a tiny portion of our mountain range, which we'll call sequence space. But there are still many, many peaks to be found in the sequence space that's been unexplored. Synthetic DNA that's explored from this uncharted territory, may have useful therapeutic functionality. We can explore the rest of the peaks to get there. While I was a medical student on the wards at Mass General across the river, I was really fortunate to work directly with some amazing rare disease patients and their families. And something that struck me during this process was this. If we were more able to precisely target the affected cells in these patients and the cell types specifically, we could have more effective therapeutics in general. Most of our genomes, in fact, are composed of regulatory DNA. This is DNA that tells genes where and when to be expressed. Cis-regulatory elements, or CREs, are one type of this DNA, and they can act like a genetic dial. So if we tune this dial, we can obtain different CREs that lead to different levels of gene expression in different tissues. So if we learn the rules of that regulatory grammar and build around them, we can find the peaks of new mountains and thereby generate a whole new world of therapies. We could engineer our DNA for any number of functions, things like higher gene expression, greater cell type specificity, greater tunability, and so on. Achieving this vision for rare disease therapeutics, of course, requires the right team, and as Anna alluded to, I'm pretty fortunate to have a great one. There are 2 major elements to achieving this next generation of rare disease therapeutics. One is designing the right CREs, and the second is getting them to where they need to go, delivery. And so in pursuit of these, I joined the Sabeti Lab and the Bhatia Lab. Pardis and Sangeeta are 2 extremely intelligent and wonderful and kind mentors. I'm very lucky to work with them. Pardis' expertise is in computational genomics, and Sangeeta works in nanotechnologies. And so these are the 2 areas that we need to bring together to make this happen. And so in the Sabeti Lab, we've come up with a way to race up the metaphorical mountains I told you about, to design new regulatory elements that can work in unprecedented ways. We use an experiment called a massively parallel reporter assay, or an MPRA, to test hundreds of thousands of CREs at once. This allows us to describe the genome's regulatory code and its grammar empirically, and then we can use computational tools to learn those rules more precisely, and then design new sequences that work even better than the old ones. We can dive a little bit more closely into this approach. First, we start with the experimental end, this massively parallel reporter assay. We take a starting DNA library of CREs. We can tag on a fluorescent reporter gene onto these CREs, and then transfect that into cell lines. And so the level of expression of our reporter gene in each cell line tells us how effective that CRE is in that context. So from these experiments, we can generate numerical activity scores for different CREs in different cell types. In this example I'm showing you, we have liver, brain, and blood. In the new age of high-performance computing, we can now use those massive amounts of experimental data that we've collected to explicitly learn the rules of regulatory grammar. These models can hone in on what parts of DNA sequences contribute to CRE activity, cell type specificity, et cetera. And so we can begin to answer questions of why a certain CRE is more active in one cell than another. And more broadly, where and how we might gain functional advantages in our CREs by modifying their sequences. So this predictive machine learning model essentially gives us a bird's eye view of the mountain range that we discussed. So this is the understanding that we use to fuel computational exploration of the remaining sequence space. With our predictive map in hand, we can guide generative AI models to design CREs with the highest reach possible in these mountains, engineering for many, many desired functions. Here's how this works. We start with sequences of unknown activity, typically random sequences. These sequences are passed to our predictive machine learning model, which obtains a baseline measure of their predicted activity. We can then use a function which looks at the whole overall sequence space, finds the peaks, and then tunes the sequences as it goes to reach them. And thereby we obtain sequences with the maximum desired function of whatever we're looking for. And this is an iterative cycle. We can then test those sequences that we've designed with MPRA again, and it starts all over. Fortunately, when we test these computationally designed CREs, we see some really promising results. And there are lots of examples I could go through, but I think this is the most striking. This is a neuronal CRE that we designed with this pipeline that we then expressed in an embryonic mouse. And early on in development, you can see that there's a little bit of blue expression in what looks like the brain. So a couple of weeks postnatal, we did dissection and see that our enhancer not only expresses specifically in the brain and not anywhere else, but localizes to the cortex, a specific anatomical region. And as any neurobiologists in the room know, cell types in the brain can be tricky. So we're very fortunate to have these results. And overall, we're really excited that our model is able to build completely synthetic, short regulatory sequences that can drive really tissue-specific expression. But we have a lot of work ahead of us to extend our models to additional cell types, and most importantly, to therapeutic applications. And so with the support of the Ladders to Cures initiative, we're now working to extend these findings to novel cures in rare diseases, and specifically in muscle cell types of skeletal, smooth, and cardiac muscle. We think these cell types are interesting because they have similar contractile functionality and similar embryonic origins, but they function in really different ways and have so many diseases that affect them. And so this is one node that we think we can target. To close, I'll just tell you about a few of the ways that we're tackling this in muscle and the ways that we think a multi-disease approach using these CREs can achieve big outcomes in rare disease. The first is in Duchenne's muscular dystrophy, or DMD. DMD is a hot topic for our rare disease community. It's caused by a mutation in the dystrophin gene, and it has devastating effects on muscular tissue, which then cause devastating systemic symptoms. Gene therapies are emerging on the market right now, which aim to deliver normal dystrophin to damaged tissues or edit native genomes directly. But one key concern with all of these is off-target effects, many of which I have put up on the slide here. And so we need to ensure that our genetic products are only expressed where they're needed. That is, off-target effects are minimized. And so we think that our CREs could prove useful in this setting. And so by combining our muscle-specific CREs with muscle-specific delivery vectors, or just using our CREs with a standard delivery vector, we could achieve some pretty interesting results. And the second is thinking more broadly about what kinds of disease cases these CREs might be useful in. As I think towards being a physician scientist, I'm always thinking about the patients first, and I really appreciated Richard's approach to that. one of the diseases I think would be really interesting to target here is hereditary hemorrhagic telangiectasia, HHT. And now HHT is a disease that affects 1 in 6,000 people. It is caused by a structural defect in a protein called endoglin, and endoglin helps support our blood vessels and keeps that structural integrity. These mutations can cause some really devastating complications. By losing structural integrity of the blood vessels, you can get things like stroke, anemia, heart failure, hemorrhage, and it's of utmost importance to try and prevent these, but there's no cure right now. The therapy here is deceptively simple. If we can upregulate endoglin only in the cells where we need to, then we could prevent these complications. And so we're really excited to continue work towards that direction. And so to finish off, I'd like to bring us back to where we started. There is so much to be explored in the sequence space we haven't covered yet. There are so many mountains left to climb. And so if we combine high-throughput experimentation, which the Broad is so good at, with computational modeling that's being enabled by all the latest advances in AI and machine learning, we can get to those next peaks and achieve this next generation of rare disease therapeutics. And so thank you so much for your time. Happy to answer any questions.
Thank you, wonderful talk. So can you comment the pros and cons of the novel DNA sequence targeting CRE and compare with current nucleic acid medicine?
Sorry, so to rephrase the question, comparing current CREs used in gene therapies as compared to our new ones?
Like current practice using RNA medicine or DNA or PNA, a lot of nucleic acid-based medicine. So can you compare your methodology with current practice?
I think the beauty of this is that you can do it all together, right? So we're generating elements that can upregulate the expression of DNA or the expression of genes. And so these can be used in combination with existing gene therapies or can be used to drive their own. And so we sort of view this as a platform as opposed to a competitor here.
Thank you.
Yeah.
Arya, do you recall a specific experience in the clinic or a patient who inspired you to go into this work?
You know, I can think of so many, but I think one of the ones that really stuck with me is when I was a first-year medical student, I rotated on the pediatric palliative care wards at Boston Children's. And there I was working with this one patient who had a very rare muscular disease that caused constant contraction. And that was the moment where I realized that the work that I had been doing in the lab, which I actually started prior to my clinical rotations, had a really direct impact. His family was just so amazing, and the care team was so dedicated to the whole disease process, trying to find cures, trying to find options that would alleviate symptoms, and more importantly, emotional well-being. And so this is sort of the picture that brought me together and said, this is something I got to work on. So thank you for the opportunity.
Absolutely. Thank you so much.
Oh,
question. And how large are these?
This is an excellent question. So it's of great importance to make sure that whatever we're designing can fit into either standard vectors or new vectors, and space is a hot commodity in this setting. The enhancers that we've designed in the study I showed you here are 200 base pairs, but I'm working on shrinking them down to 50, which is about the size of the minimal promoters that we use in standard applications currently.
Brilliant. Thank you so much, Arya. I think you can all see why I'm so wildly proud. You know, if this is our future, then we have a lot to be proud of and look forward to. So as was evident in the question that I just asked Arya, one of the most fundamental ways in which we communicate, well before we had written text, for example, to communicate with one another, is actually storytelling. And it turns out that storytelling is what we've been doing here today, scientific as well as personal and family storytelling. And it's a really effective way for humans to motivate one another. It's a way for us to motivate our elected officials, for example, our colleagues, our community members, our neighbors. And so the next speaker is actually a master at storytelling. I think that maybe you have heard of Effie Parks and her quite well-known podcast series, Once Upon a Gene. But if you haven't, I think she's here to tell us about, there she is, about this journey, really, that has been inspired by her son, Ford. So I will let her share the rest of the story with you. Effie.
to the Broad. Thank you to the Broad and Ladders to Cure and the Termeer Foundation, and especially Jillian and Katie for inviting me and for just being so great. So hi everyone, like Anna said, my name is Effie Parks, and I'm so excited for this 10-year anniversary event. It's such an honor to be here with you today. I always have a little bit of writer's block before I do a talk like this. I'm always maybe a little bit down on myself, and I don't have any letters behind my name. I'm Ford's mom, and I know I'm not alone there. I can stare at a blank page for weeks and sip my coffee. I will call a friend and stare some more, look for ideas and advice, and I just hope for some inspo to shake me. And then it happens. The dam opens up, and the ideas that were stuck behind blank pages and negative self-talk come flooding out. And that happened the other day for me. I was like, wait a minute. I'm about to speak to a group of some of the most evolved scientists, some of the most brilliant minds, people who lead experiments, who find patterns, who create assays, people who analyze genomes for breakfast. Surely these people believe in maybe the existence of little gray or green dudes that are light years away. So why were aliens my idea for this talk that opened the floodgates? It's because being a rare disease parent is actually like being on another planet. But let me back up and take you to my story. 10 years ago in 2015, I married the love of my life, and I got pregnant with my first child. It was everything I had dreamed of, a family, motherhood. Everything was as bright as a supernova. We even had a space-themed nursery, because the obvious next step for my child was NASA. The metaphor really did feel magical, and a perfect child ready to explore the infinite universe of possibility was just magical. But when my son was born, it turned out that he was on a different mission. And had I listened to the doctor's explanation, maybe a mission that would never have even left the launch pad. It was certainly one that we did not expect. Something was wrong with our son, and we didn't know what. We found ourselves fighting to simply keep him alive for the first couple of years, addressing symptoms with hardly a moment to look up and recognize where we were. And then genetic testing pinpointed a diagnosis of an ultra-rare disorder called CTNNB1 syndrome. At 16 months old, it felt like we had crash-landed onto an alien planet. There was no roadmap, no research, no data points, just emptiness. There were no humans around us that spoke this new language, and there were no treatments, very few patients, and no one to tell us what was next. In a moment, our world had gone from the infinite hope of the night sky to the vast, cold emptiness of space. Back then, just 10 years ago, social media was not the vast resource for families like mine like it is today. Costs for genetic testing were sky high, doctors were more skeptical, and the pace of research felt painfully slow and progress impossible day to day. We were desperate, alone, and searching for a guidepost and any community that might say, hey, we're here in this weird place too. We were not travelers here, my family and I. We were pioneers set out to terraform this strange place and to make it a place that we could live. And so I realized that if such a community did exist, I better go looking. So I did what any rational person would do, and I started a podcast. And I radioed for help. once upon a gene became my version of an interstellar radio beacon, broadcasting into the void, hoping someone, anyone, would respond. And they did. Suddenly, families like mine were on the same frequency. We were all these little spaceships floating out there in the void, pew-pewing meteors and space monsters disguised as insurance denials and feeding tubes. Trying to dock, compare notes, and find refuge. For many of us, it was the first time that we realized we weren't alone. What started as my voice and the laugh track of my son, it quickly multiplied. We compared notes, we shared tips, we found a new sense of comfort, hope, and a new sense of urgency from each other's stories. In these early days, we learned from trailblazers, families and advocates who had been fighting for decades on behalf of rare diseases they'd discover long before the rest of us showed up. They passed on that knowledge and that share your homework mentality with each new org that showed up. These pioneers, they taught us how to navigate the medical system, how to push for research, how to speak up at conferences like this, and how to find the essential resources we needed simply at home. And the coolest thing, with each new family on board, our community didn't just grow in numbers, it grew in power. Maybe we weren't just a bunch of lone stars in the sky, and maybe we were a constellation. We were building momentum and amplifying each other's voices. And it's incredible to look back now and see how this turned into a movement, with parents and caregivers driving research forward, funding studies, partnering with biotech and research and industry. We might not have lab coats, but we know our children better than anyone, and our insight is crucial to any progress. A few advances of the last decade, once described as far off, has rapidly become part of our reality. There's still plenty of work ahead, but every year brings new tools, new knowledge, and most importantly, hope. Genetic testing is a diagnostic tool. Once was the cost of a mortgage, in my son's case, a brand new car. Now it's less than a Netflix membership. And you can get it through telehealth on your couch, just like Netflix. This means that families can get an answer fast and skip the black hole of being or misdiagnosed. They can get to work a lot faster in accelerating global collaboration among advocacy and companies, scientists. Patients and caregivers know that we shouldn't have to have our data locked away in a silo that we cannot access. We know to pool our resources in orgs like Combined Brain, and we innovate. Telehealth and remote care technology have been a godsend. No more flying across the country just to see the one expert. For the last year, 40 of our CT and NB1 patients have been wearing a device that's been tracking their movement disorder in preparation for a clinical trial. These rare families are more than mom and pop nonprofits. They fund grants, they found biotechs, they have matured into CEOs, policy experts, and they radiate love for their children and their community like a sun. The shift is happening. Each year, more families connect, and more solutions inch closer to reality. Solutions that once sounded like science fiction. And now there's the hope part. We didn't cling to hope because it sounds nice. Hope is prickly and sharp, just as it is warm. We build hope because it is fuel in our rocket, the thing that keeps us searching for answers. Every new technology, every new data sharing platform, every connection that we make is a shooting star. And in moving forward together, how do we keep that momentum going? We collaborate relentlessly. Families, scientists, biotech, academia, healthcare providers, and policymakers all have to work in sync, because progress in rare disease thrives and can only work in synergy, not silos. We tell our stories, because personal narratives don't just inspire hope, they open doors to funding and research and understanding. And because it turns out people actually do want to hear them. We question everything, since we can assume any system works until it doesn't work until it truly does. We push for new approaches, new cures, and clinical trial designs that mirror our real life experiences. And finally, we stay hopeful, not the fluffy, blind optimism kind, but the belief backed by action, and maybe a little bit of rare parent crazy eyes. Because at the end of the day, that's how breakthroughs happen. So there you have it, my slightly derailed journey from starry-eyed new mom to space-themed nursery to crash landing in an alien world of rare disease. In the end, though, we realized we weren't in this galaxy alone. We formed a collective, we fostered community, and we turned heartbreak into innovation and hope. And that's the kind of unstoppable energy that we're here today celebrating. When your world blows up, you adapt, you innovate, and you share, and together we keep going while pew-pewing all of those evil space monsters. So thank you for letting me beam my story into your orbit today. Each of you are so vital to this unfolding mission. I'm convinced that together we can end rare disease. While I have felt like an alien on another planet these last several years, and I felt like I was forced onto that planet, I am starting to understand that maybe we were never actually needing to board ships and rocket off to some distant galaxy to find our people. We were all right here on Earth, scattered by isolation and circumstance. But once we connected and once we found each other, we realized that this planet has everything we need to innovate, collaborate, and make a difference. This isn't about chasing the stars. This is about bringing the stranded explorers home. There will be setbacks and failures on the way to the safe landing that we all dream of, but we cannot accept. What we cannot accept is progress being stalled by the lack of collaboration, communication, and willingness to work together. I want to hear everyone, our scientific community, academic industry partners, stepping up and sharing hope and knowledge, just like our rare disease community has done. We don't have to separate to a whole new world. We just have to join forces to make this one better than we found it. In all seriousness though, if anyone does have intel on some extraterrestrial beings, please find me after, because I'm pretty sure I could pitch them to join our patient registry, or at least give me a sample for the biobank. So beam me up, Brodies. I could do this all day with the analogies. Thank you, I'm done. Thank you for listening. If you have a question, I'm happy to answer. I know we're running a little over time.
Questions for Effie?
Okay, thank you.
Thank you so much. Okay, so by now you're noticing how masterfully Jillian has been weaving in the stories this afternoon. So we're doing science, and then we're doing advocate in family stories and then coming back to science. And so it's my great pleasure to bring us back to some really cool, innovative ideas around another rare disease, Marfan syndrome. Our speaker is Avanthi Raghavan, who is a postdoc in Patrick Ellinor's group. She's also a cardiologist at Mass General. And again, one of the people in the younger generation will wildly proud of, and you're about to find out why. So, Avanthi.
Thank you so much for that introduction, and a huge thank you to Ladders to Cures as well as the Termeer Foundation for organizing this. It's a real privilege to speak here today. So as Dr. Greka mentioned, I have dual roles. I'm a cardiologist at Mass General, and then I'm also a researcher here at the Broad. And really the focus of all of my work, my clinical and my research work, is thoracic aortic disease. And a few years ago, that led me to a rare disease called Marfan syndrome, which is a Mendelian form of aortic disease. And so for the next 10 minutes or so, I want to tell you about some of the work we've been doing to accelerate therapeutic development for this disorder. So what is Marfan syndrome? Like I mentioned, it's a Mendelian connective tissue disorder. And it's caused by mutations in the fibrillin-1 gene. So fibrillin-1 encodes an extracellular matrix protein. It's expressed throughout the body. And so as you can imagine, these patients have multi-system connective tissue issues. And often they're recognizable because they're unusually tall and thin. But over the course of their life, they develop many issues, including skeletal issues, pulmonary issues, ocular issues. And then most importantly, the most feared complication of Marfan syndrome is aortic disease. So what is the aorta? It's the biggest blood vessel in the body. So every year, our hearts beat 35 million times, and with each of those heartbeats, our aortas have to expand to accommodate that entire blood volume under high pressures and distribute blood to all of our body's organs. And so that takes a lot of contractile as well as elastic strength. And so in Marfan syndrome, fibrillin mutations lead to aneurysmal degeneration and weakening of the aortic wall. So over time, as early as adolescence into early adulthood, individuals develop aneurysms or enlargement of the aorta, and this can predispose to acute complications such as aortic tears or rupture. And this, in fact, is the leading cause of death in Marfan syndrome. So traditionally, there have been two pillars of treatment for Marfan. So the first is surgery. You can have an open heart surgery where the surgeon goes in and basically cuts out the diseased portion of the aorta and replaces it with a graft. And this may or may not be curative. For some patients it is. Other patients, they continue to have issues over the course of their life. They continue to develop issues involving parts of their aorta that were not replaced. And so for those patients, we can offer medical therapy. And really, we only have two classes of medications we can offer these patients. They're both blood pressure agents. And they show sort of variable benefit in terms of slowing aortic growth. But the one thing I want to point out is that both these classes of medications have been around for decades. And so I think that kind of speaks to the fact that there really hasn't been much meaningful therapeutic advancement in this area for many, many years. And so when I first became interested in studying aortic disease a few years ago, I noticed that traditionally genomics and a lot of these high throughput technologies that we routinely use at the Broad had not ever really been applied to aortic disease in a meaningful way. And so our kind of overarching goal was to think about how we could leverage some of the strengths of the Broad Institute in genome-wide association studies, single-cell multi-omic studies, high-content imaging, and combine them with more traditional approaches such as mouse modeling and molecular biology to accelerate the pace of therapeutic target identification and development for Marfan syndrome. So over the next 10 minutes, I hope to give you a flavor of some of the efforts that we've been doing on several of these fronts. So one of the first things I wanted to do is I realized we didn't have a tissue biobank for Marfan syndrome. And I was extraordinarily lucky to work with a couple of folks. Antonio Cresso is a cardiac surgeon at MGH, as well as Ben Landis, who's a pediatric cardiologist at Indiana University. And we actually established a pipeline for collecting aortic tissue from Marfan syndrome patients. And so what we do is we chart review and we consent all eligible patients, not only with Marfan, but many other rare genetic aortopathies. And so we consent them prior to their elective aortic operation. And then we go to the OR, and so as soon as that aortic tissue becomes available, we flash freeze it so it's as fresh as possible. And then we bank it. And just in the past few years alone, we've been lucky that MGH is such a high volume aortic center. We've collected 16 tissues from Marfan syndrome patients to date, as well as over 30 healthy controlled tissues. And so in the next few months and years, our goal is to use these tissues to inform various techniques, including single-cell transcriptomics and epigenomics. But flash frozen tissue just kind of gives you this snapshot of gene expression at a certain moment in time. And so what we really wanted is we wanted a system that kind of gives you a more dynamic readout, right? So what if we want to perform a drug screen? What if we want to perform a CRISPR screen? So most of the pathology in Marfan syndrome really centers on the smooth muscle cell. The smooth muscle cell is the most abundant cell type in the aorta. And I mentioned earlier that the aorta needs a lot of contractile strength, and it's the smooth muscle cell that gives it that contractile strength. And so it turns out that if you have a fresh aortic biopsy, you can actually isolate that muscular layer, and you can put it into culture, and the smooth muscle cells grow out, so you can establish lines directly from patients. And so we've been doing that over the past few years. We've generated multiple Marfan syndrome lines from multiple patients. And so then we asked, can we develop some sort of morphologic profiling technique in order to kind of understand how these disease cell lines look? And so to do that, we took advantage of something that we know about end-stage aneurysm. So in end-stage aneurysm, smooth muscle cells lose a lot of their contractile abilities, and they upregulate a lot of synthetic markers like collagen. And so we stained a couple of different components of this cell, the actin cytoskeleton, collagen, as well as SMAD, which is an important signaling mediator in the TGF-beta pathway. And we just looked at our cell lines under the microscope. And so here I have two different wild-type lines on top, and the bottom I have a Marfan line. And if you look at like the highest seeding density, the cells are just totally different, which is not surprising. So the wild-type lines are green, they have lots of contractile features. And then the Marfan lines, they're more orange, so they have all this collagen. And in fact, when you look at these morphologic features in the disease lines, over a third of those features are totally different in the disease line. And so we're really excited to see that phenotype. And of course, we can quantify individual phenotypes, but ultimately our goal is to identify features that are common across multiple donor lines. And eventually, the goal really is to perform a reversion screen, so to look for small molecules or to look for guide RNAs that can actually revert that disease phenotype right back to a healthy cell. So I've spent some time telling you about things that we want to do in order to accelerate therapeutic development. And so for the second part of this talk, I actually want to tell you a specific story about a target that we've already found and that we're super excited about. And this is a really special story from our lab. A few years ago, our group did a genome-wide association study looking at aortic size and found over 80 loci that were associated with aortic size. And I have a Manhattan plot from this study showing all those genetic loci with the p-value on the y-axis. And I simplified it to show you just two loci. So one of them was fibrillin 1. So we found fibrillin 1, which is good. That's reassuring. And then another gene that jumped out to us very early on was this one locus on chromosome 16 called WWP2. And that kind of made us raise our eyebrows because we'd never heard of this gene being connected to aortic biology before. So what was it about this gene that made us excited? Now, when we do a GWAS, of course, we found lots and lots of genes, but really what we're looking for is the unicorn. So we're looking for that one gene that has some set of intrinsic qualities that tells us this gene is going to be successful in the target development pipeline. And WWP2 just had them all. So what were those features? So first of all, when you look at expression of WWP2 across all tissues in the body, what jumps out at you is that WWP2 is highly enriched in arterial tissues, and the expression is in fact highest in aorta, which tells you that if you were to employ a WWP2-directed therapy, theoretically it should have the highest impact in aorta, which is what we want. And then the second thing we found was a very favorable directionality. So we found that the minor allele of the lead SNP at this locus is associated with less WWP2 expression in aorta, as well as smaller aortic size. So what does that mean? Less WWP2 equals smaller aorta, which is good, right? So that tells you first that we should focus on developing a WWP2 inhibitor. And two, right, it's easier to develop an inhibitor typically to a protein product than it is to develop an activator. So for all these reasons, the strong genetic association, the tissue specificity, and this favorable directionality, we felt that all the stars had aligned, and this was the target that we wanted to go after. So how did we do this? We used mouse model. So it turns out that there's a Marfan syndrome mouse model. It has a point mutation in fibrillin 1, and it phenocopies the human disease beautifully. So these mice are long, just like Marfan syndrome patients are tall. They have skeletal overgrowth, and they develop aortic aneurysms from a really young age. And so we took a mouse that had WWP2 inactivated alleles, and we crossed it onto the Marfan syndrome background. And then we looked at the size of the aorta using ultrasound. So we actually do this in humans, except a mouse is about 25 times smaller. So we can use ultrasound to look at the aorta in mice, and I've kind of colored in the aorta on those mouse images for you. And what we found is that from wild type in purple to Marfan in blue, you see an increase in aortic size, which is what you would expect. And then you see that that increase is attenuated when you go from the Marfan to the Marfan mice that have WWP2 inactivated. And then when you look at those aortas under the microscope, they look better. So one of the histologic features of aneurysm is you have a lot of elastin fragmentation. We clearly see that in the Marfan mice. And that seems to be attenuated to some degree in the Marfan mice with the WWP2 inactivation. So WWP2 is an E3 ubiquitin ligase. It functions to transfer ubiquitin to a substrate protein. And so in some ongoing work, we're using different proteomics approaches to identify downstream substrates of WWP2 that may be responsible for this effect on aortic biology. So that kind of brings me to the end of my talk. And I covered a lot of ground today, but what I want to say is that a lot of this work is early. We started many of these efforts just a few years ago. And our long-term goal is really to integrate functional genomics, high-throughput biology, as well as mouse modeling to identify novel therapeutic targets for Marfan syndrome. We've invested a lot of effort the past few years into resource development. So we're establishing a biobank for Marfan. We're establishing a lot of primary cell lines from these patients. And then, of course, we've identified a promising target already that we're really excited about studying further. So thank you so much for your attention. A huge thank you to the Ellinor Lab for making all of this work possible, as well as to our collaborators. Thank you.
Hey, Avanthi. Really cool talk. Just on the human genetics, is there a natural experiment where you have a Marfan's patient who just has a natural mutation in WWP2 that happens to have a small aorta or something that's protected? Is there like a proof of genetics there?
Yeah, so that's a great question. When our group did the original GWAS, we did look for rare variants. This wasn't in Marfan patients, just in general. So we looked for rare variants in WWP2. Unfortunately, the burden of rare variants in WWP2 just wasn't high enough to understand if potentially there's some kind of protective effect. Certainly in the future, I think it would be really interesting to look at maybe large cohorts of patients with Marfan syndrome. There is a lot of phenotypic heterogeneity in Marfan syndrome, so there are over 3,000 mutations in the fibrillin 1 gene, and different mutations are associated with different disease penetrance and expressivity. So it may be a little bit challenging to sort of tease apart the effect of the mutation versus the effect of WWP2 inactivation, but it's a really, really excellent question.
Thank you, fantastic talk. So in your GWAS map, so besides WWP2, there are a lot of top hits.
Yes.
So what are those genes, and why do you study those?
Sorry?
Why do you choose WWP2, not all other top hits?
Oh, why it was WWP2? Yes. So I think WWP2 was just, of all the 80-something loci that we identified, that was the one that we thought, just based on a certain set of qualities, just seemed most likely to be successful in just from a drug development perspective. Obviously, you don't know until you model it, but I think we were really excited about the clear tissue specificity for aorta, as well as the fact that it seemed like inhibiting WWP2 was the way to kind of attenuate aortic disease. So that kind of jumped out just from the genetics data, but I think ultimately our goal is to look at a lot of these other GWAS loci using some of these using CRISPR screens and other screens with our morphologic profiling platform to identify other potential targets for the disease.
Thank you.
Thank you.
Our next talk is going to take us back to the company creation space. So I'm really happy to introduce Tinashe Chandauka. He is a 2024 Termeer Fellow. He's a physician and serial entrepreneur. Every time I talk to him, I learn about a new company that he has spun out from his work. He's currently the CEO of TMAB Therapeutics, which focuses in the cancer space, but today he's going to talk to us about his journey with Nandi, which focuses on cancer, autoimmune, and rare disease. Welcome.
Okay, thank you. Okay, no. You can do it at the end. You can do it at the end. You can do it at the end. Let's get the device. Let's see, is this working? I'm not screwing it up. Great. How are you guys doing? Are we sounding like people who are solving for rare diseases or not? Yes. Can I hear the people at the back? Can I get a hell yeah? There we go, there we go. So, over the past 2 weeks, I have been in multiple cities. I've been in Houston, Texas, where we're building some of our exciting companies. I've been in Florida, in Fort Lauderdale, having a great time with one of my co-founders, working intensively on a manufacturing problem in China. And then I'm here today. Why do I do this? I would do nothing else with my life. I love creating companies, finding people who have a mission that they want to use biotechnology to solve, and empowering them to do that. That is my gift on earth, is to bring people together and to solve those problems. I'm an MD, PhD by background. I was a Rhodes Scholar at in Oxford University. I was born in South Africa, my home. I love that place. Haven't been in 5 years because I'm so busy doing this stuff, but it lives inside me. And it's important for you to know that, because the people in Africa need these medicines. And my thesis is, if I can create medicines that can translate into something valuable for all the people in this room, then we'll cover my continent. So that's the thesis. That's the thesis of impact right there. I'm very lucky that I went through a background of having studied at Oxford and then been involved in venture capital, working for Europe's largest early stage life science fund called Oxford Science Innovation. I was involved in helping to stand up 55 life science companies out of the company's 100 portfolio companies. That equals very little sleep. And you know what? I put my hand up for all the tough things, because that's how you become a good company creator. And because of that, I became involved in the Oxford vaccine. Now, some of you didn't get that, but 2 billion people on the planet did, and it did make a difference for them. And I was very proud to have been one of 2 people in our life science team who put my hand up in January of 2022 to say 2020, to say that we would shut down the company because the flu vaccine had failed. And at the bottom of everyone's screen was this little pulmonary infection that's happening in Wuhan in China. It's not a big thing. And it turned out to have been one of the most important projects I was involved in. Through that, I was recruited by a gentleman called Bobak Azamian, or Bobby Azamian, to work as Director of Business Development at his biotechnology company in California. And that afforded me the opportunity to work in a public biotech company from 40 employees to a much bigger level. I could sit in meetings where we were acquiring companies. I could work on everyday problems like, how are we going to make this app that reads people's eyes to launch a drug product? But all of these things have been instrumental in becoming a company creator. I want to stop here for a second and highlight the woman who's here. She is my sister. And I managed to convince her to leave her job at Meta. She's a corporate lawyer by background, M&A lawyer, trained at Baker McKenzie, multiple IPOs on the London Stock Exchange and New York Stock Exchange, and worked at Morgan Stanley and then later on at Meta. And she acts as the executive chair of Nandi Life Sciences, and I'll tell you a bit about Nandi today. We're values-driven people. Every day we're making decisions in our companies that are, yes, based on science, yes, based on financials, but that are based on integrity and who we are. I'm a doctor first, which means I have a line. There are things that I will not do. And I think it's important for everyone in this room to think about that for a second. Sometimes there are medicines we can develop, and sometimes there are medicines we should not develop. And it comes down to these values. And you'll notice that the first one there is being relentless, because developing drugs, like the family that's here, requires you to give everything of yourself. Think about that for a second in terms of what that means. But also to be an advocate, to be impactful, to be empathetic, and sometimes to actually take one for the team, because you have to make decisions that are guided by integrity. And I think all too often we don't talk about what that actually means. Look at me. You know exactly that I don't sound like I come from here, and you know, like I'm in a country where sometimes things can get a little bit weird at this moment, let's be honest. And so what did I do? I knew that I wasn't gonna raise money here because I didn't go to Harvard, I don't have the social network, let's just be honest. So what did I do? I found a group of crazy people in Texas who have a lot of money and believe in crazy people like me to build something like this. And what did they do? They gave me healthcare benefits, a stipend to work at, and that's the Texas Medical Center, right? So please throw away all your conceptions of who is capable to innovate, where they live. You know, just hustle, just do what you need to. So I found my path to being a company creator in Houston, Texas. Beautiful place. The world's largest life science community, actually. If you arrive there, you'll see it. It's a city. They see 10 million patients a year. Rare diseases are pretty common down there, and great doctors who trained here and around the world who are there doing great things. So let's just work together. So what do I do? At Nandi, what we do is we partner with scientists with great ideas who generally may not know someone who has a lot of money because maybe they didn't go to college with them, or they just don't know how to communicate it fluently. And what we do is we will spend months landscaping a particular disease area, identifying unmet clinical needs. For example, what if you have a renal kidney disorder and we've just understood the basic biology, but there are no medicines to treat that? What if a child suffers from seizures and we've just begun to understand the biological targets, but it's not a sexy subject that VCs are investing in right now because it's not a validated target? Those are the situations that we like to get involved in, where we've understood the basic biology. When we sit down and we do the financial models, over $700 million of peak sales opportunities, places where antibody therapeutics and other protein therapeutics could make a difference. This has to do with manufacturing. We can talk about that someday. And also where we know that we have IP, so freedom to operate. You'd be surprised how few things meet those criteria. But if we can do that, and then most importantly, if I can stand in front of you and actually define the disease condition we're going to be treating, so many good ideas cannot translate because what are you heading to, right? So often starting off with the disease area, the problem, and then heading upstream like the family here is doing, helps us to always remember the patients, right? That we're going to develop a drug that doesn't give people additional adverse effects. So what have we done in the last 2.5 years? Also very little sleep. We've built 4 companies. Most of them are stealth, so I can't tell you exactly what they do, but I'm very excited because 3 of them will be announced this year. These companies have raised over $50 million, largely led by our most advanced company. We've partnered with a Boston venture capital fund. I wish I could, I want to say thank you to one of the gentlemen there, but when he watches this talk, he'll know it. And by the summer of this year, it'll be announced. Let's start from the back. So we will often find a concept, 6 months to 18 months. We will front the cash to in-license the technology, to build the company, to find consultants, to find team members. That means that my family liquidated most of our assets because I had a crazy dream. And when you start to try and build something, good luck finding someone who'll give you money. You often have to front it. So when I moved from California to Texas, a lot of things were lost. A lot of sacrifices were made. And so, you know, on the back of that, my wonderful family and friends, we've been able to find these technologies to build them in-house and take this forward. Then when we decide to pull the trigger, when we understand that we even have conversations with pharma far in advance, understanding who will buy this company and take it to the world. Once we are sure that we can meet those criteria, once we know who the customer is and that I speak to patients, to physicians, and they said, yeah, if the drug did even half of that, yeah, I'll take it. Then we really start to go on the road to find real money. And you know what's more difficult than finding money? great talent that's going to run it. Because, they actually ask tough questions. They're like, so I've got my great job at Sanofi or Pfizer or something. Why should I leave that great job and come work with you? Have you figured out the science? What are the risks? Walk me through it. Nah, that's not true. Go look at that. Go look at this. So I'm very excited that for our companies that we build, the conversations are very intensive. It's often over dinner, but generally people say yes, this is something they want to get involved in. I've jumped into the helm of TMAP Therapeutics, an oncology company, a partnership with MD Anderson as its co-founder and CEO. Let's dive deeper into that as an example of what happens. Do you understand what is happening here? Who knows? Please, could you share? Sorry? There we go. It's finishing your chemotherapy course. We hope you've beaten it, but it's a big moment. So the scientists here, Professor Anil Sood and Jishwan Anne, identified that there were escape pathways to anti-VEGF drugs. And anti-VEGF drugs are anti-angiogenic agents that stop blood vessel growth into solid tumors. They can also be used in the eye. But the problem is they haven't generated overall survival benefit. They've generated progression-free survival benefit, which means that VCs won't fund that. Because unless if you can demonstrate that a single drug in a trial beats everything else and can demonstrate overall survival benefit, they're just not going to fund it, right? Because otherwise it's Russian roulette. And we can't do that with pension fund money. So these two scientists worked for years trying to understand how women with ovarian cancer, which is a rare disease, needed new therapeutics that could demonstrate overall survival benefit in that field. And despite the fancy drugs we come up with, some of you in this room were involved in that, PARP inhibitors that are used in 50% of women with ovarian cancer eligible for that. Despite the great new antibody drug conjugates that have been developed that only qualify for 30 to 40% of the women who have recurrent platinum ovarian cancer, we still have this huge group of women who have platinum-resistant ovarian cancer. And you know what's common across all the lines of therapy? That not-so-sexy drug that doesn't do much, what people say, bevacizumab, very important drug. And when you speak to the physicians, they say, it's a great drug. We use it in some women for 9 to 18 months. We can keep using it when all the other drugs fail. What can we do about it? But I go and speak to VCs and they say they don't care about it. So last year, after these scientists have been working on this for 15 years, they published in Nature a phenomenal publication demonstrating that the reason why CA-125 goes up when patients are now on bevacizumab under their continuation treatment in recurrent ovarian cancer is because of these two novel secreted ligands that are overexpressed, very similar to VEGF. And they mapped out the transcriptional pathways that are involved in these being overexpressed and leading to new blood vasculature growth and immune cells growing. And what we've been doing as a company is building multi-specific antibodies that can target anti-VEGF pathways while targeting anti-CD5L and anti-EGFR6 and stimulating an immune response. Over the next coming weeks, you'll see us more in Boston. There'll be an announcement about what we're doing. But I want to say thank you to our competitors who have paved the way. Summit Therapeutics and Akeso have developed a bispecific that's going to transform the cancer space, anti-VEGF plus anti-PD-1. It's demonstrating 50% progression-free survival benefit versus pembrolizumab, which is just astonishing, in a phase 3 head-to-head trial in China. And MD Anderson is helping to fast-track the approval of that drug in the United States by running a large phase 3. So what do? We found talented people. Heren, who headed up antibody engineering and discovery at AstraZeneca, came out of retirement. His wife is not very happy with him. And he helps us late at night to engineer our tri-specific antibodies. Rakesh, who's like a mentor and dad to me, he used to head up animal studies at AstraZeneca in oncology. And he acts like a mentor. He makes sure I don't do dumb things. And he's our fractional chief scientific officer. We also have a ton of partners who work with storied companies like Johnson & Johnson, Argenx, and Genmab. I can't tell you who those are, but they're really good at what they do. And yet I still stress them out because of my demands. And this is what we're dreaming of. We're dreaming that if we can raise $50 million over the next 24 months, we'll be able to run 2 phase 1 trials at MD Anderson, one with our super Avastin drug, which combines CD5L, VEGF, and EGFR6 in tumors that respond to anti-VEGF drugs but need an additional kick of immune activation. That's what CD5L does. Makes macrophages start to make inflammatory cytokines, it recruits T cells. With our other multi-specific antibody construct that has a checkpoint inhibitor component to it, we want to target cancers that have responded to checkpoint inhibitors but could need an additional boost. Things like microsatellite stable colorectal cancer, where we've had a lot of difficulty. So that's it in a nutshell. We could apply it to all of these cancers, and this is why we get the companies to phase 1, we start the relationships with pharma, and then we say, we can't do this as a small biotech. The right thing for patients is to hand over the technology to great teams that can run the trials around the world and get these drugs to patients as quickly as possible. We do the numbers. This is an example of the peak sales opportunity for a tri-specific construct that would be looking at non-small cell lung cancer. And that's it. I think what I want to show you is we're very busy. We're building teams for our other portfolio companies. And this is the tough thing. If you want to learn how to build a company, please speak to me. Happy to be your friend. And maybe one day when you do it, I'll be there cheering you on. And we can work together through this structure. So thank you very much for your time. These are all the wonderful people who help us and all the partners who've made it possible, who took a bet on our company, gave us massive discounts to get going. And that's the truth. That's how these things start, is people take a bet on you. So thank you so much for your time. No questions. Good.
Our next talk is going to shift our lens a little bit. So we are going to hear from Bart, who is from the Netherlands. And he's going to talk to us a bit more about how he uses AI to facilitate both care delivery and drug development through his work in the Netherlands. So we'll get to see a little bit about how he thinks about how an academic medical center can fit into all of the innovations that we've heard about today.
Thank you, Catherine, and what a wonderful program you've developed. I'm so proud to be part of it, and a little bit anxious to be following such great speeches. And what I want to present today is really, of course, AI as a sort of a catalyst through which can enable a healthcare system to adopt all this great research and all these great companies and this development into something that actually reaches the patients at scale. So I want to talk about an enabling ecosystem. And as mentioned, I'm from the Netherlands, so it's closest to my home to talk about the Dutch ecosystem. I'll elaborate a bit about that. So I'm currently employed by the University Medical Center in Groningen. My main role there is to develop our corporate AI strategy, which covers both the medical side, but also the research and the educational sides. I'm also the president of the Dutch Clinical Research Foundation, and I'll talk a little bit about that as well, because it's an important step to this enabling ecosystem. I also want to talk a little bit about more the European perspective or the larger European ecosystem, and finally dive into a few examples of the applications of AI in a medical system or in a hospital. So first of all, the Dutch Clinical Research Foundation, which I currently chair. If you want to develop new agents and bring them into healthcare, of course, research is a key component. And in the Dutch Clinical Research Foundation, we really align partners in research to make sure that the process is as efficient as possible. So first of all, there's the researchers themselves or the clinicians themselves. The Dutch academic hospitals and the larger hospital systems, they're all collaborating together in the DCRF. We also have industry, so the innovative pharma, the CROs are also part of the foundation. Our regulators, so the Central Committee for Human-Bound Research, the Dutch Medical Ethics Committees, and also a research funding entity. And they're part of a larger connected ecosystem. And that's how we see the Netherlands as well. It's a really condensed, small country. Geographically, it inhabits about 18 million people, but it's really well connected. It's well connected in terms of data infrastructures. It's also well connected in terms of physical infrastructures. And it's really central to Europe. We also house the European Medicines Agency as well as the European Patent Office. So some anxiety exists around Europe being a really difficult place to bring your research to, especially if you want to scale, because it's, of course, not one country. It is many different countries. But what is the interesting thing about that is that in Europe, we have a habit of collaboration because of the fact that we have many different countries in one legal economic entity. So on the left, you see that in general, more and more commercial research is being done in single countries. So one country and all your eggs in that one basket, let's say. Especially with China, but also here in the US, it is a more common practice. And over time, you see a trend that it becomes more common. Whereas in Europe, increasingly, we are doing multinational studies, especially since the introduction of the clinical trial regulation. That has been much more the case. And also, if you look at the total volume of studies, continuously the European Economic entity area has been well positioned to do multi-country studies. And increasingly, there are advanced therapies in those studies. So over 15% of these studies are using advanced therapies. And I think that's good news for the rare disease space. Because I think nowhere else it is more common that advanced therapies are used as in the rare disease space. And there's a clear reason why. And this is a hashtag that was introduced in Europe a few years ago. It says 30 million reasons, which represents the 30 million patients of rare diseases in Europe. And actually some studies show that it might be as high as 36 million. And that's twice the population size of the Netherlands. And these patients can't wait. So this is something that we really need to focus on. And one of the great ways to do that in a European context is through the European reference networks. I don't know if you've heard about these before, but the European reference networks bring together patients, healthcare institutions, academia, around certain rare diseases. And in the Netherlands, we've been really focused on promoting participation in the European reference networks. So out of the 24 that were established in 2017, 7 are actually coordinated by Dutch specialists, and we participate in nearly all of them. And it's a really fantastic way of knowledge sharing and expertise sharing, and it really allows us to quickly go to a pan-European collaboration. On a more Dutch scale, we have also invested in Rare NL. And Rare NL is an organization that brings together academia, healthcare professionals, but also industry around rare diseases and drug repurposing. So this is a hub really for the development where we work together from these different disciplines. Some of the work that was done, I won't go into much detail there, sorry. So this Dutch ecosystem, the way it works is we have our university medical centers, there are 7, and this is really a system of collaboration that we have. Over 80,000 people are employed in those institutions, about 12 billion turnover every year, most of the science actually is developed there. Most of the rare disease specialists are there. And then there is also other infrastructures. So for instance, Health Array is really about aligning data infrastructures across the nation to make sure that we have those data that we need to do research and innovation. And also we have some different organizations such as RareNL, but also the Dutch Center for RNA Therapeutics, which you may be familiar with, who really support the work in this rare disease space. Now, some of the examples from a more clinical approach, and this is not something that we yet do in rare diseases space, but we are sort of trying to see how, especially generative AI, is helping our healthcare system. And those examples, I think we can later expand into the rare disease space. So if you look at AI in a single hospital, the way we approach it when we look at this technology, we think of different value streams that we can use this technology. The first one being efficiency, which is quite easy to identify. So use AI to reduce administrative burden, for instance. Then AI for discovery. I think a lot of the work that's being done, especially here in the Broad Institute, is about AI for discovery, or the machine learning or the deep learning that's already been explored for many decades, but also now with the generative AI. Then the AI for precision, I think that's where it becomes more interesting, because, for instance, we use a project that's called the Pharmacogenetic Passport, where through gene testing and multimodal data approaches, we can find and identify unique therapies for individual patients, and AI is really supporting us there. And the last one is AI for augmentation, and there I think you should really think about also the patient's perspective in this one. So imagine you being at your physician, he or she is telling you all about your disease, your therapy, and the things that are ahead of you, and AI can really help us by, for instance, taking that information and then relaying it to the patient in a really understandable and easy accessible language, for instance. So we're actually doing a lot of work on that, and I'll skip this one. So what we've done is we've really developed a framework in our healthcare institution. We've looked at what are the unique challenges that generative AI bring to us in terms of, for instance, biased medication. We heard hallucinations or confabulations in one of the previous presentations as well. But also things like liability. How does it work if such a system fails or if it makes mistakes? So we've generated this framework. We've developed a team that brings together all the key expertises that we need. And based on that, we've done a pipeline of different use cases that we've introduced. So that's, for instance, helping our doctors getting a draft response to the patient's questions, but also an AI text assistant that helps doctors draft patient-accessible language in their discharge notes, and some other functionalities. So we currently have 8 functionalities in clinic today. And then the next frontier for us is no more typing, and that's really about the ambient listening, so listening into the clinical conversation, moving that into high contextual text and then into high quality data. And I think therefore the rare disease space is going to really open up, because if we are able to do that, go from a discussion in clinic into high contextual text, into high quality data, we can start exploring for new avenues to identify patients, for instance, through symptoms that they describe during those conversations. Of course, this is all being done in a context where it's really important that we also consider the ethical, legal, and societal aspects. And for that reason, we've established the ELSA AI Lab, which focuses on this specific part of the introduction of this technology. And there we really want to understand if, for instance, you only ask a lawyer whether something can happen, or if you only ask somebody who is really involved in privacy aspects, then the answer might be too black and white. And if we can do that from a more framework perspective on the ethical, legal, and societal aspects, I think a lot more can be done with that data. And I think that also opens up the patient's perspective from this technology. And the reason I mention that, so in Groningen, we actually have an amyloidosis expertise center. And when you look at patients that come in with this destructive disease, which is really rare, by the way, One of the very common complaints is that they have is numb feet. And there are so many reasons why you could have numb feet. But if you look at family history, for instance, patients with amyloidosis typically have a strong genetic predisposition, so they will have a lot of history in their family with such complaints. And if that could only help us to more quickly identify and diagnose those patients and ensure that the damage that is done by this disease is captured early, then I think we have a lot to win. So I have one last, and I'm looking at the time, one last example that I wanted to give, and that's the Genome Med for All. And actually, this is the work that's done, a follow-up project that was done by Eduard van Beers from the Utrecht University Medical Center. So that's another hospital. And what they have actually been able to do is to model through federated learning, through synthetic data, something for sickle cell disease that enables them to identify new patients on a quicker scale. And this is led by the Euro BloodNet, which is one of the ERNs. And I think that shows that there's really hope for those patients in those rare diseases. And maybe still a last one. Last month, a paper was published in Nature Medicine about Solve-RD. And this is a network where actually from about 6,500 patients that had undiagnosed rare diseases, they brought together, pulled together a lot of data. They pulled together expertise from different European reference networks. And then through this project, they reevaluated those 6,500 patients and were able to identify over 500 patients or diagnose over 500 patients with a rare disease. And I think that goes to show that technology really helps us to move forward in the rare disease space. So to briefly summarize, and I hope I've been able to relay my message properly, I'm not trying to promote the Netherlands here, but I think it's good to look at it from an ecosystem perspective. So how, from all that important research, how do we actually bring it to patient? And that's really through a collaboration in the ecosystem, both in a national perspective, but also from a European perspective. The drug repurposing was mentioned a few times during the presentations, and in the Netherlands we really feel that that's a really high potential because it brings together to the patient therapies today rather than 10 years down the line, which I don't want to make an argument for that we stop doing that innovative work, but look at what we already have. I think we should really get experience in using those AI models in healthcare in general, so not just the most advanced models, but also everyday applications. So for instance, browsing through patient data to find new patterns. And lastly, and I think this is something where the Massachusetts area and the Netherlands have already, have a long-standing relation. This model of collaboration, which is really part of the DNA of this ecosystem, and it's also very much the DNA of our ecosystem in the Netherlands. So leading by example, I think, is a key work. And with that, I want to thank you for your attention.
Questions for Bart? I have a question. I'm curious about your impressions from your vantage point as to the potential for collaboration between the EMA and the FDA in the context of rare diseases. What have you seen? What do you see as challenges or opportunities in terms of those interactions?
Yeah, thank you for the question. And I don't know the extent to which these organizations already collaborate. I know that what really helps, of course, if, let's say, the ecosystems themselves collaborate and that the regulators could follow. Over the past years, I've seen positive developments in terms, if you look at, for instance, from the US side, the ARPA-H initiative, which really fosters collaboration between the transatlantic collaborations. On the EU side, the same has happened, right? So especially in the health research space, there's opportunities for US counterparts to collaborate with EU counterparts. And I think if we can, foster that collaboration much more, then I think those regulators will follow. I don't know whether they are already in advanced state in current discussions.
Okay.
Okay, thank you.
So we've gotten to sort of the final part of the day. Should we just do this as a standing panel for a shorter time? How are my co-panelists thinking about this? Is it okay? Okay. We have some mics. We'll pass them around. If you could all just join me up here, and if it's okay to have you stand. And Effie, of course. Wow, what an afternoon. I know it's been long, but I think fascinating, and I just want to give, again, all of the speakers a round of applause for just incredible stories and scientific discoveries. Clearly, there's a lot to be hopeful for and a lot more work, of course, ahead of us. So I know we had like this whole long sort of list of questions that we wanted to discuss, but I also want to keep us on time. So I just wanted to maybe, just have a conversation and just think about, whoever, feels like sort of jumping in. From everything that we've heard today, or from all the different, groups and constituencies that you represent, different stakeholders in the field, what would you point as to a recent win, something that you're really sort of positive about, each from your different vantage points, and whoever, wants to jump in first. What can we name as a recent win?
I have a couple. I'm going to start with CTNMB1, the disorder that my son has. Two things. The CTNMB1 Connect and Cure Foundation here in the USA, obviously from fundraising from families and generous donors around the world, has funded Michelle Jacob Lab at Tufts, who has partnered with the Broad and was awarded the NCATS NIH Trend program. So they will be working to optimize a lead compound for the disorder my son has. And second, our CTNMB1 Foundation in Slovenia is well on their way. They've manufactured our AAV9 gene therapy and hoping to go to clinical trials this fall. And to shout out some amazing rare advocates out there, Terry Piravalakis just appeared on Kelly Clarkson. Sunita Malapati was at the White House speaking, and I just think it's so profound that these rare advocates are showing up and raising their voice in places that maybe they don't belong. I'm at the Broad. I mean, people are listening to us, and it's moving, and it's really wonderful, and I'm seeing that these aren't just diseases anymore to people, and that people are seeing that there's a community and human beings behind it. So I'm hopeful, and I think the future is bright.
I love it. Yes. Of course.
So I talked a lot about patient insights. I want to share one win for one of the partners that we're working with, Global Genes RareX. They are a patient registry platform. They were recently awarded a 1.5 million technology grant from the Termeer Fellow Foundation. We're also a grantee from the ZZI grant, so we're very happy for both of them. They're essentially using AI to better understand patient data, and that is so powerful because a lot of times it's really hard for the patient, the families, to fill out pages and hours of survey. They have all the information for years of history, but it's just so complicated to share all that data with your clinician, with the researchers. I'm really excited to see AI in general being a tool to capture more patient insights, data, stories as part of research.
Fascinating. Yes, I think that's incredible, and there are many opportunities ahead. Any more takers? Recent wins? Any takes? Okay, fine. We'll kind of move into the next topic. I was just curious about, we talked a lot today. I mean, there was a mix of scientific and sort of more stories from inspired by your work in advocacy, working with family foundations. But the thing that struck me as a common theme, has been the tools. There's a need for tools. Sometimes there's scientific tools. We discussed the discovery of some of those, and other times there's other tools. So, and then that brings us, of course, to who has access to these tools, and how do we make it better. I know that many of us today talked about, problems with access, how it's unique that we can all be here in Boston together, talking about these topics, but, others who are listening to us and sort of thinking about all the things that we discussed today, does it sound like something that would be accessible to them? So I'm curious about how you're thinking about this from your different perspectives, and maybe, what are the tools that you're focusing on and how you might be securing their access to others in our broader communities. Any takers?
Well, one of the reasons I'm doing my research at the Broad is just this enthusiasm for making publicly available tools, not just the software, not just the assays and molecular techniques, but also putting data into databases and making them shareable and doing our very best to make them usable by everyone, researchers, families, clinicians as well. So to me, that's a really important part of being here.
Maybe another dimension on the tools question. I think obviously the Broad does a lot of work in developing innovative tools. I hope one of the themes we've heard today is how much having end application, a rare disease, or something that you're really working towards can help focus your efforts so that the tool is something that can be extensible, can be applied. Because I think, in my day-to-day work at Thermo Fisher, we do see lots of innovative tools come in, but not all of them have clear applications that can or are worth being democratized because there's only a few people at the Broad who could possibly use the tool. So part of it, I think, is really partnering with patients, with patient advocacy groups, or partnering with the end users of these tools to really understand kind of how applicable these are, and having some sort of focus really helps drive to usefulness and ease of democratization.
Sorry.
Go ahead, please.
Okay. I was also going to say that places like Simon Searchlight and Combined Brain, again, are working to gather these patients and really think about where the similarities are more than the differences, and really keeping a pulse on the extra motivated advocates out there.
Yeah, and just to layer on what you both said, I think the intentionality is really critical. So all our contracts, our mission is really provide that access, and we'll figure out how to do it. But let's start with the assumption that we must do that. So for example, we share all the data back immediately for research use. We don't blind anything. We don't take the best hits off the top. Everything gets shared back because we want to advance that research, and it may not be us doing it, and that's okay. And so kind of letting go of that protective aspect that sometimes has, at least historically, been in the space, that's a decision that we made. We're not going to do that. And we won't get it right every time, but at least having that kind of guiding principle that, we are in this together and we're going to solve it together. Let's start with that, and then we can figure out what we do along the way. That's where all the tools and databases and all that kind of take place.
And maybe just one more thing to add on there, since we are in a more academic setting here. One of the things we heard when we first started this rare disease journey was how many rare disease groups have partnered with academia to develop models, whether that's mouse models, iPSCs, et cetera, and how many of them had difficulty just getting the models out of the university, developed assets, hits, targets, et cetera. And I think sometimes the tech transfer offices, the powers that be, view these assets as having substantial value before they've been moved out of the institution. But the reality that anyone in biotech will tell you is, once you take it out, you still need to plug tens of millions of dollars into it before it has any value at all, and it may not be worth anything too. So I think there's this embedded assumption sometimes that every asset has huge value coming out of the institution, and there's a big incentive to just not let things out, but that substantially slows progress, both in innovation, both in democratizing access, and then also for our patients ultimately.
And I want to follow up on that because I think that's sort of a theme for the Termeer Fellows, which I greatly admire in all of you. But this idea of jumping into starting a company or becoming a CEO of a company where there's not a proven business model in the classical sense. So would you like to speak a little bit about that?
Yeah, so one of the things that we get dinged on by VCs is, why are you changing so many things at once? Why not just one thing? And the whole point is, if you want a substantial change, it's probably not going to be done by any one little change.
And so a huge credit to the Termeer Foundation for taking a chance at putting me into the 2024 fellow group. It's been fantastic, and I have to say the mentors are really key along the way. I think Tinashe also mentioned his mentor outside of the program, where they're taking a chance on you. It's not on your technology or your business, or somebody thinks that you can change whatever it is that you're proposing. And all the Termeer mentors, for example, go in with that assumption, and there's not this like knee-jerk, well, that's not how it's done now. Whereas if I talk to VCs, it's like every other conversation. It's like, that's not how it's done. Like, well, yeah, and look at the results. The idea is let's try something different. It may not work, right? It's not guaranteeing that, but the only guarantee of not doing anything different is you're gonna get the same thing out. And so that's where that mentorship and all that comes in. So absolutely that the ecosystem driving that support for just take that shot and see if it works. I think it's key.
And Tinashe said, the greatest challenge is, finding people, right? Like-minded people, building those teams. So I'm curious about, both from, anyone's perspective here, really, how hard has it been to find, you told us some elegant stories about how, your podcast, for example, or how you have done, you have reached out to other families to try to build community, but what are the challenges in terms of finding people, either people to work in your startup or people to work with your foundation, or both? In some cases they're intertwined. So how are those challenges, and how did you overcome them?
Yeah, I'd say for Ada and I, it was probably a lot easier than it was for Effie a decade ago. We're lucky that there's institutions like the Broad, institutions like Effie's podcast, and support from organizations like Ultragenics, who have a boot camp to bring people together. So in a lot of ways, the only reason Ada and I have been able to accelerate so quickly is because of all the groundwork and foundation that's been led. Now, there are still challenges. I think the preclinical path for these rare diseases and ultra-rare diseases is a lot more clear. There are still a lot of questions to be solved in the clinical and commercial stages, obviously, whether that's manufacturing or trials, what have you. Many different questions still remain, but at the very least, the front end of it, the network is there, and there's a more clear roadmap than, of course, when you started.
Add another point that may not be directly answering the question, but I've been thinking about that. For our community, we're still trying to identify more families, more patients out there. Our disease was discovered in 2016. We have 300 families identified since then, and they're mostly children. And what we're trying to find are more families out there, and with the use of AI, mapping facial phenotypes to the disease. When I was listening to your talk, I was very inspired to think that can we use tools and technologies to find more families out there, not just in the US, but around the world in an easy way, so we can expand our community, and then our community becomes our partner in this journey. So I think there are many ways we can accelerate even finding families in our community as part of the equation.
And maybe just one more point building off of that and connecting to the access point you made earlier. Early last year, Ada and I were lucky to have our story and Luki's story on the front page of the Boston Globe. Through that, a mom of another ZTTK patient reached out to us. They're the only other ZTTK family we know of in the state of Massachusetts. They live in Springfield, Massachusetts, and they only got their diagnosis last year. Their son is age 12. So I'd say around access, they're only a 2-hour drive from Boston, one of the best, medical and scientific centers in the world, and yet their access to these technologies is so limited. In a lot of ways, even in America, someone in a rural or exurb area of America may have less access to whole exome and genome sequencing than someone in a major city like Istanbul or something like that. So we really need more access to these tests. There's a lot of research out there about how it's estimated 20% of cerebral palsy patients actually have a genetic disease of some sort. Like, these are all very obvious low-hanging fruit to increase testing and just help patients. Obviously, we then need to solve the problem of finding treatments and cures for them, but identifying them is like the very first problem that needs to be solved, and that will really highlight the problem.
Yeah, I'll piggyback off both of those comments, because what I was going to say is we need to look at the patients who have passed this. They're at the hospital and have young kids, and people are worried and wondering what's wrong. But what about those grown kids, those adults that have autism, epilepsy, and cerebral palsy, who were totally missed? Over 35%, it's believed, maybe even up to 50%, is that especially the patients with cerebral palsy have a genetic etiology. And so how do we get those genetic tests to those people with those umbrella diagnoses? I think we'd find a lot of rare disease patients.
Makes a lot of good sense, and maybe part of the reason why there should be efforts that really, health and rare diseases and pediatric diseases affect every state, every constituency all around the country, and of course, every country around the world. So I hear your point about sort of access and making sure that that's... So now, for the last question for each of you, we are transported. We just celebrated 10 years for this gathering that we do every year here at Broad. We've been doing it for the last 3 years successfully with the Termeer Foundation. Again, very grateful for that. Now we are being transported somehow through a spaceship, perhaps. To 10 years from now. And I want each of you to tell me the one thing that, we have learned or achieved, or what does it look like in terms of when you look out there in terms of the rare disease landscape from each of your different vantage points, 10 years from now. We don't have to go in order, but if anyone has any first takers. Yes?
All right. I'll say precision medicine is what I want to see happen. In our talk, we talk a lot about the right drug for the right patient at the right time, and let's bring back the right dose at the right route. So I really believe that precision medicine, hopefully it's less than 10 years, can really transform how we think about treating and supporting each individual based on their needs, because it's not a one-size-fits-all, as we all know. So I am really excited to see where this goes and how we can get the data for that one individual and really tailor the drug to that specific person.
That's great. Any more visions 10 years from now?
Maybe another dimension of that, more from an industry perspective, and another area where perhaps academia can work more with industry. There's a lot of innovative modalities that have been innovated by the Broad and other institutions, the cell therapies, the gene therapies. As anyone who's spent time in the space knows, one of the biggest challenges in these spaces is manufacturing, right? There's promising science, but we just can't get the manufacturing right. I would like to see a world where academics work more with industry further in advance to solve those challenges along with the science, because those challenges can be just as important to the success of these therapies, and I'd like to see those solved much better in the future.
Absolutely.
I think it's not unreasonable within 10 years to imagine just a much more systematic approach. So whenever any baby is born, they're sequenced and any issue is immediately diagnosed, at least solving the genetic component of this. And then if I'm allowed a second wish, then,
as many as you want.
Then at the very least, can we get the low-hanging fruit of matching up the existing drugs with existing disorders using some of the kinds of technologies we talked about today? If we can just do a first pass of all of those against each other, we really would get far.
So what you're saying is let's maximize the drugs that we have already invested billions in building, and make sure that they're used for every last patient who can benefit from those. I think that makes very good sense. Although hopefully in 10 years we can also be on our way to some new treatments, right? to Ada's point. Anyone else? Vision for 10 years from now? I'm
so
really
a lot
curious.
of this has been addressed, but there's concept of the systems level approach, and I think it's a systems level problem. It's not just a genetic problem; it's also access to healthcare, access to a lot of other factors. We work with some families in remote South Africa. It is a whole nother problem. You're dealing with that, right?
Sure.
And the patient here could just go to Boston Children's same day, be seen by the world's top experts. In this case, that's just not possible. So what do you do? And the answer is not nothing. It's something. But now, what is it, right? And so I think my vision is really that systems approach. Like, we're all in this together. Rare isn't rare. It's just fragmented. And I think recognizing that is going to be a very key acknowledgement of the problem and the sheer scale of the problem that we're facing. And it's a system that we need to build together. So I hopefully in 10 years, you have the same event, and
Yes.
we have 500 successes, and then, well, we don't really need to do anything more. That's my vision for
we're all out of work.
for this 10 years
That
from
will
now.
be great.
Yeah, that's right.
10 years, we're all out of work. That's a great message. Any more visions?
Similar to yours, I would love to see newborn screening and whole genome sequencing on every baby, not just a baby in the NICU, not just a baby with symptoms, to completely eliminate that diagnostic odyssey that causes so many problems for everyone involved. So that's what I would like to see, and I think that is closer than 10 years, but who knows with bureaucracy, so that's my wish.
That's great. Any more? Anything to share, Nathan, or you're covered by, that sounds great. Well, so I'd like, I think, to close this out. You know, it's just, again, a huge thanks to all of you for coming, to all of our speakers for excellent talks, really on a broad array of topics. I want to thank again Katie Ligori, who's been sort of the logistical organizer behind all of this, and my right-hand person, and Dr. Jillian Shaw, who has curated an incredible program. So thank you, Jillian. Really, I want to thank all of you. Big And just to say, sort of as a closing, kind of as a closing argument, if you will, I think we've done a lot of good work over the last 10 years, and of course, much longer than that for all of those who have been in this space for a very long time. There's clearly a lot more work to be done. But I think that today you can see why I'm a glass half full optimist. I think that there's a lot to be hopeful for. We heard great science, promising technologies, also very interesting approaches, and a huge amount of commitment from all the stakeholders in this space, the patients, the families, the advocates, the scientists, the physicians, the biotech and pharma leaders who are committed to this area. So I think there's a lot of work ahead, but there's a lot of hope that there will be great progress coming our way. If we continue to think about this as a space that we own together as shared sort of stakeholders of this space. And so I'd like to end with the idea that it will take each and every one of us and all of us working together to make progress. So thank you again, and please enjoy networking reception outside after this. Thank you again.